participants. We have our speaker, Dr. Sandhya Gupta ma'am from NDPGR. So let me give you her brief biodata. So I will share my screen so that you also can visually see her biodata. Okay. I hope it's visible, ma'am.
Yes.
Okay. So our today's speaker, Dr. Sandhya Gupta from Tissue Culture and Cryopreservation Unit, with her specialization in economic botany, will be our today's guest. And she is currently principal scientist and office in charge at this TCCU division. And her salient achievements and nature of work involves in vitro conservation and cryopreservation of temperate and minor tropical fruit species, in vitro collection that includes 316 accessions of 10 genera. She has worked on many more. And she is also a curator, and as curator of the cropland, she has introduced about more than 200 new exotic accessions in the country from USA in the year 2005. She is also a faculty of IRI PG school, and also has served as a resource person in Bioversity ICAR Center of Excellence on in vitro conservation and cryopreservation. And she also owns a number of awards like Biotechnology Overseas Associateship Award from Department of Biotechnology, Government of India. Then she is also one of the fellows of Indian Society of Plant Genetic Resources, New Delhi, Indian Society of Horticultural Research and Development, Uttarakhand, and also vice chair of a commission on plant genetic resources during the year 2014 to 18 in the International Society of Horticultural Sciences, Belgium, and also awarded ISHS Medal Award in 2018. And her division is really doing great work in the area of tissue culture and cryopreservation. And regularly we are seeing very good number of publications coming from her division. So ma'am, I want to congratulate you for that. And with this brief introduction, I request you to please continue with today's session. Thank you, ma'am. I will stop sharing the screen now so that you can share.
Okay. Thank you, Mamta, for such a generous introduction. And I would be happy to tell again that this year again I have got elected for ISHS vice chair for plant genetic resources. Yeah,
congratulations,
so
ma'am.
thank you. I need to update my biodata. I will do that. I will do that. Yes. So I'll share my slide. It's visible?
Yes, ma'am, it's visible.
Okay. So can you see it now?
Yeah, now it's presentation mode.
Okay, yes, presentation. So good morning, all the participants, and welcome for the first session of this training. I mean, today, this presentation was around 11 o'clock, but I said that if the slot is available, so I got it pre-born. Probably that is the reason people are not able to
no, ma'am.
connect.
No, it's not the reason, ma'am. Already, yeah, it had to start from 9:30 only.
Okay.
Yeah.
Okay. So the topic which has been given to me is application of tissue culture techniques for genetic resources management. I'm going to talk about plant genetic resources. So, plant genetic resources, you have been going through many lectures by now, and you know how important plant genetic resources are for us and for future. Conservation is very important for them because they are the basic building block of any crop improvement program. But tissue culture techniques are for the management, they add into the management of plant genetic resources. And you can manage it, but the tissue culture technique has more advantages where you do not have proper or method to preserve them. You must have, in other speakers must have covered it, that the plant genetic resources, there are many crops which produce seeds, orthodox seeds, which can be dehydrated and can be conserved in the seed gene bank. But there are many species which do not bear seeds, or they are non-orthodox seeds, meaning they are recalcitrant seed having high moisture content. And those seeds and vegetatively propagated crops, as well as there are many other species where problem occurs, the seeds are not viable or they are not available. So those crops can be conserved through tissue culture techniques. I'm sure that if you have not hands-on practice on tissue culture, but you must have read about it. So the tissue culture started in 1902 with the basic principle of totipotency. Heberland has given the principle of
slides are
totipotency.
not moving.
I have not moved the slides yet.
Okay, ma'am. Okay.
Yeah. So I'm just giving the introduction of tissue culture, that from where it is started. Started way back in 1902, where Heberland used tissue culture. He is also called as father of tissue culture. And he gave the term totipotency. Totipotency means any cell of the plant can regenerate and give rise to a full plant. So we later on used this principle and this method for management of genetic resources. So you can see tissue culture techniques can be used starting from germplasm collection, multiplication, germplasm exchange, disease eradication, and germplasm conservation. So these are the main aspects of plant PGR management. And NBPGR is the nodal institute who is involved in managing the plant genetic resources for food and agriculture. It starts from exploration, and there the material is being collected, and then these are being characterized. After that, being conserved, documented, and in between they are also checked for their health, so that they are free from any contaminant or any disease. And further, at the last, these are being conserved, and everything is being documented in the form of database. So I will take you through one by one all the aspects of where tissue culture has been used. So first and foremost, I would like to tell you that how important the culture establishment is. Everything depends on this basic, you can say, method where you should know how the culture, in vitro culture. So first and foremost is germplasm acquisition, that you are collecting the germplasm from the authentic source, and it is with the correct identification, the genus, species, from where you are collecting. So these are all involved or include into the passport details. So once you have the passport details and the authentic source and species identification, you can go further. And depending on the plant, you can choose the propagule. That this can be shoot apices from the vegetatively propagated crops or from the tree, wherever you are. And you have to decide. It can be a node, root, leaves, vegetative bud, and embryos, etc. These could be the explant for your culture establishment. Then once you have taken this explant, you get to surface sterilize it, because in the field, the plants have, or the propagules have dust and some of the any like spores or fungus or insects in insects eggs. To remove all of that, you use different sterilization techniques and different sterilization material. It can be ethanol, 75% ethanol we use surfactant like soap solution, and mercury chloride also we are using in other countries. Somehow it is banned, but we are still sometimes use. Otherwise, most probably we are using, most frequently we are using sodium hypochlorite and bevestine as a fungicide, and we give treatment. So starting from the germplasm or accession, we need to, when we develop the protocol, this particular step is very important, so that in later conditions, we do not have any contamination in the culture, and the cultures are contamination free. So once the culture is established, then a suitable media, culture media, which is can be find out from the literature if it is available, or if it is not, then we have to develop the how much hormone concentration it should be, how much combination of different hormones are to be used. This we need to standardize. So there are several culture medium. Mostly it is used Murashige and Skoog medium, which is being which has microelements and the macroelements in it. Also, we add sucrose so that the plant has all the essential ingredients for their proper growth. So cultures are culture medium. There are many. Murashige and Skoog is one. Then B5, WPM, Gamborg media. There are several media which has been given by several scientists for different crops. There are some specific crop-specific media, or there can be Murashige Skoog media normally works for many crops, but for woody tree species, WPM media is preferred. And once we are putting cultures into culture media and we check for bacterial contamination, the method is on nutrient agar we are using, and we are doing the streaking method. If the bacterial contamination is there, probably we first need to remove that bacteria. If it is endogenous bacteria, the method is that you grow out the material in the field and take the fresh explant. But if it is a bacterial contamination and if by sterilization it goes away, then it is fair enough. After that, we also ascertain the virus status, because viruses are the one which you can't see by naked eyes. So you have to do ELISA and all for checking the virus. If it is virus free, then in tissue culture, you can multiply it. If it is not virus free, then there are methods to remove the virus or get the virus free plant. So it is preferred for tissue culture that we take the virus free material or propagule for getting them into the culture. After this, once the culture is established, they are moved to the multiplication media. A multiplication medium has little high concentration of growth hormones like BAP. This is cytokinins, BAP to IP, and it can be multiplied. So this whole method is one procedure which is very important for this plant establishment and plant multiplication. This all is a one protocol like micropropagation, and micropropagation is prerequisite for conservation or mass propagation. So one should have the whole protocol. The micropropagation has 4 steps: that culture initiation, culture establishment, multiplication, rooting, and hardening. This makes a full protocol. And once you have the full protocol up till hardening, you can go about for conservation, cryopreservation, and everything, so that you can regenerate that plant and keep this plant in tissue culture, multiply it, as well as you can transfer into the field as and when is required. So micropropagation is the key technique which is required for any further intervention. So first, if you can see this, how the tissue culture techniques are being used in different plant genetic resources management activities, the germplasm collection is the first and foremost. There are several, when we go for exploration, there are several material which are bulky and they cannot be transported like that. Plus, sometimes there are very less amount of plant material or propagule, which you need to, and exploration is sometimes takes 15 days, 20 days. So it is preferred that you take the germplasm and put them, sometimes if it is seed or a fruit where seeds are embedded in the fruit, you can bring the fruits and extract the seeds and do the thing. But if sometimes the vegetative propagule are there, they can be, they can be rotten when you reach to your lab. So the best way to conserve them or to save them is that you take the material along with you, a medium along with you, and take the material, have the propagule, and put them in culture media. So culture media, they can survive for say about 1 or 2 weeks without damaging the tissue. So it is a direct placing of plant tissue on the culture media for transportation to a distant laboratory for further multiplication. And once you reach to your laboratory, you can multiply those. If in between, so the steps are that you collect the propagule and give them sterilization method. I mean, some of the sterilization treatment, so that the fungus and bacteria, when you reach after 2 weeks in your lab, so contamination doesn't come. So the fungicide or surface sterilant, you give those treatment, and after that, this is just kept in the tubes or whatever jar and brought back to the lab. Sometimes wild species, endangered species, these are the propagule, or they are the preferred material where we can use this technique. This method acts as a support activity for germplasm collection. As I said, that if you have seeds or fruits, you can bring them, or if you have cuttings, you can also bring them to the lab. But if you don't have those, then you can use the tissue culture method, and it's transferred to a laboratory where further experimentation will be carried out. So the germplasm is safe, and even if it is a very small quantity, you can bring it to your lab and multiply it. That is not a problem. So these are the steps that you select appropriate tissue for culturing. If it is a bulbous crop, you have to choose bulb. If it is a dicot, you can use shoot tips, or you can have the axillary bud or nodal explant. So like that. And cutting the tissue in appropriate size, because you can see the vial. This is the document which IPGRA has. IPGRA is now Biodiversity International. So way back, they have given a guidelines like in vitro collecting techniques. And here you can see the vials. So these kind of vials you can take, and with the media, and it doesn't take much space. So you can take maybe 25, 50 vials, and it doesn't take in a small box, and you can go to the field and take the material with you have to bring those small equipment like the forceps, scalpel, blade, and sterilant with you for sterilization. And you can go in the field, and removal of any adhering soil, you can wash it, disinfect. Disinfection and surface sterilization, as I said in that slide, that you can give that treatment. And damaged tissue, I mean, it is understood that you should not put damaged tissues in the vial. So these should be healthy, green material and suitable culture media. When you go for exploration, generally you know for which crop you are going for exploration. So you know a little bit idea from the literature or from your experience that which kind of media is required for it. And generally prefer that little more agar should be put in the media so that it is hardened and it doesn't, it is not a loose kind of, otherwise if it is a very loose media, then it will spill from the bottle. So we add little extra agar so that media is solidified, and then your tissue is firmly placed into the media till you reach to your lab. An inoculation onto suitable culture media, then transferring the material to suitable incubation chamber. So once you come back, you can process this material. You are bringing your material safely here. If in the lab you see that some of the contamination has come, so you can again treat this plant, this material, because it is still in the condition that can be taken for the further sterilization. So again, maybe 1 or 2 minutes of sodium hypochlorite you can put, and or you can give bevacizine treatment under the laminar flow, or you can go about further. Then tissue culture is also very, very important for germplasm exchange. Exchange meaning import and export of germplasm. And basically for vegetatively propagated crops, because seeds can be exported and imported without any problem. Put it in a pouch and you can exchange it, and they withstand 15-20 days time without facing any problem. But when vegetatively propagated crops come into the picture, they are perishable, and these are very difficult to transport sometimes in a healthy form, and sometimes it is very bulky, so difficult to send across the from one place to other place or across the country. Then the bulbs, grafts, seedlings, these are the propagules where they don't withstand. If your consignment is at the higher temperature sometimes and left unattended, then those propagules, those this, and it takes a lot of money to exchange the material from bringing it in India or sending it to different countries. It takes a lot of money, so we need to really take care of that the material which we are importing or we are exporting should reach to the indenter in a very healthy condition. So tissue culture really plays a very good role in that, and careful packing is here. You can see the material, in vitro material has gone to a different country, and this is how it is packed. These star packs are being used by USDA. From USA, we are bringing in vitro germplasm, so they use this kind of plastic packets where media is there, and they put little material, and it doesn't take much of the space, and it can be safely transported. There, it doesn't have any any possibility of breaking anything, because when we are transporting it, and preferably we are transporting sending the material across in a plastic vial. That is safer. But even if they are culture tubes, then we have the packaging, proper packaging, so that they reach to the destination without breaking them. And this INIBAP uses this kind of similar kind of packets. Then, as I said, why we use in vitro cultures for germplasm exchange? Excuse me. Because when material is in vitro condition, it is free from insect paste and pathogen, except the virus. And as I said, that viruses are the one which we cannot see from the naked eye. But other, like fungal spores and the pathogen, like the bacteria, we can see from the naked eye, and we can take care that when we are exchanging the material, those material is free from insect paste and pathogens. Quarantine regulation. You must have had, I think Dr. Dubey has given you overall view that how the quarantine regulation work for sending the material out or the importing. So those quarantine regulations, they are not very stringent for when the material is in vitro. So it is easy moving of material if it is in vitro condition. Then germplasm distribution becomes easier because the material is in the form of small plantlet, which can be transferred. Or if it is a rooted plant, then you can directly send it and acclimatize it, and it can be put into the field after acclimatization for a few weeks, and can be directly transferred to the field. So that is also an advantage, and it can be easily multiplied. If sometimes the requisition comes that they want some material. So how to go about it? If we have the extra material, then we can, from there we can share. But if we don't have, we ask for the time, and we can just multiply the material and give few tubes to the indenter. It is very easy. And sometimes the indenter doesn't have the tissue culture method, the tissue culture lab with them. Then what we do is multiply our plants, root them, and acclimatize, and we can give the potted plants to the indenter. So the distribution becomes really easy with the intervention of tissue culture methods and survive. So the material, as I said earlier, that if your prepigule is in the test tube or in the small vial on the media, it can remain healthy and healthy at the ambient temperature. You don't need to have a low temperature or a culture room or something. It can be okay for 2-3 weeks, and then by the time the material will reach to the indenter safely. So next comes in vitro germplasm conservation. By now you know the seeds. As I said earlier, that seeds are there for seed gene bank, but vegetative propagules are being conserved in the field. That is a conventional way of conserving them. But then we also give emphasis on duplication of safety duplication. So when the material of vegetatively propagated crops where the seeds are not a proper way of conserving, because orthodox seeds, as you know, are heterozygous and they are cross-pollinated crops. So heterozygosity does not give you true to typeness of the. So when you you need to do the genotype conservation, then tissue culture is must. So those all the germplasm are being conserved in the in vitro gene bank. And there are several methods how we can use. The first and foremost is the culture establishment. So once you have culture established and multiplied, there are several methods by those using one or the combination of those techniques. You can we can go for in vitro conservation. These methods involve a reduction in temperature and light, and use of growth retardants and growth regulator, use of osmotic regulator, type of enclosure, size and type of culture vessel, and induction of storage organ. So I will take you to the different examples of each one of these methods one by one. So it is a slow growth or normal growth strategies which we are, as I said, that the tissue culture gene is very important for many crops. And in NBPGR, we have seed gene bank, in vitro gene bank, and cryo gene bank. So in vitro gene bank is using tissue culture method. So by changing physical parameter, we can slow down metabolic activity of the culture, and it does not multiply that much. For temperate crops, it is advisable that they can be stored at low temperature below 10 degrees centigrade. We have several temperate crops, many medicinal plants, they are temperate. Many temperate crops like strawberry, pear, apple, peaches, these are also can be stored at the low temperature. So maybe 4 to 10 degrees centigrade. 4-5 degrees also is very good. I'm going to show you the experiment which we have done. And for tropical crops like banana, its temperature is 10 to 20 degrees centigrade because it is a tropical crop. And other tropical crops where the temperature could be more than 20 degrees centigrade. So these tropical crops, we are conserving them in the culture room with the standard culture growth condition. So you can see these are here. This example is shoots of chestnut. 48 months of storage, they are healthy and at 8 degrees centigrade. So depending on the crop which you are dealing with, you can choose the temperature, whether they can be stored at the low temperature or they can be stored in the normal culture room temperature. In USA, they are conserving, they are working on, different labs work on different crop groups. So crop group which is, they are working on soft fruits and berries. They're conserving it in slow growth. By reducing the light, they are conserving it on the basal media without any hormone and reduced light and the temperature. So this is in combination of light and temperature. You can conserve and take the shelf life of cultures for a longer period of time. We did an experiment with the Frigeria ananasa and Frigeria vesca. Frigeria ananasa is a strawberry. You all know about it. And see, these cultures are capped at 25 degrees centigrade in the normal culture intent. So after 6 months, you see how much growth is there. So this is very good for multiplication, and these cultures can be multiplied, and from one tube we can make maybe 20 or 30 tubes. But here, when we put those cultures at 3 to 5 degrees centigrade in dark, then see the growth. Though all the cultures are very healthy, but the growth is reduced. So the effect of low temperature and the low light is that the metabolic activity is reduced. And by reducing the metabolic activity, we reduce the growth of these plants and increasing the shelf life of plant. So that is the purpose. And these cultures, we can see that then go. Here you can start, this browning has started. So after a few months, this whole culture will go away. So we need to subculturate. So when the culture condition becomes like this, or here again, see, you can see little less brown. And here it is a very dark brown. So we will take this material. So it will go more than 6 months. This is Frigaria vesca, and these two are Frigaria ananasa. These are Frigaria vesca. So this is how we know. Otherwise, you will think that how will we know that it is time to subculture? This Frigaria ananasa is good enough. It can go further. So we every 2 months we take an observation, and when the culture condition start deteriorating, we subculture it. Subculture meaning is we take the fresh media and make a small plant, reduce the leaves and all, and take the auxiliary, depending on the, in Frigaria, the propagules are here below. That's why I'm saying again and again. But in dicots, you have got a shoot like this, and then you can cut from the internode and have a single node you can transfer to the fresh media, and it can grow and give you a fresh plants, new plants. So growth retardants can be used, malic hydrazide, abscisic acid, psychoside. There are several which can be used to reduce the growth of culture. But normally, these are not preferred for long-term conservation. Like in our repository, we do not use any of those growth retardant because these are said to be inducing the genetic instability. So that is why what we are using is reducing the growth regulators in the culture media. When we reduce the growth regulator, that means reducing the growth of the metabolic activity, and so the growth doesn't happen that much. But when we have to multiply, we increase the cytokinin in the media and multiply. Depending on your objective, you use the media. And use of osmotic regulators. These are also like sucrose, mannitol. These are also used as osmotica, where you add them into the media, and the growth of your culture is reduced. So in fact, this work has been done by Dr. Neelam Sharma was here. She was working, and work was done by her. Effect of mannitol on in vitro propagation and conservation of Bacopa manneri. This is a medicinal plant. Then type of enclosure. Earlier we were using cotton plugs. In cotton plugs, on the tube like enclosure, the disadvantage was that the media used to dry up very quickly. So we switched to polypropylene cap. So any kind of container you can take, and the capping enclosure you can take. So we saw that type of enclosure is very important for conservation purpose. So we used to do the cotton plug first, and then we shifted to polypropylene cap. And in polypropylene cap, we saw these are the all we have done in our lab. That's why I'm telling you. In polypropylene cap, the problem was that after several autoclaving, they used to become loose. And when they are loose, there are contaminants which could be enter into the spores, sometimes a mite. So the cultures got contaminated. That is why we switched to screw cap enclosures. And the screw cap enclosure, these are the tubes which have the screw cap the caps which have a screw in it, and then it reduces the entry of spores. So the contamination got reduced to a large extent. In the type of enclosure, there is no hard and fast rule that which kind of enclosure this culture vessel you are using. There are several. The jar, glass jar, plastic jar. These are we call them magenta box. These are square boxes of plastic, and this is good for multiplying the material. But the disadvantage is if the contamination comes, the whole lot of the material goes away. But the steel test tubes are the best one because you have only one explant in there and one plant. If contamination comes, you can discard it or salvage this one particular plant, and other material remains the same. Then the growth storage organs also you can induce in the media. These are the crops where these bulblets and microrhizomes and microtubers you can grow. Like in ginger, we use Dr. Tyagi. This paper is from Husnarayan Tyagi. Dr. Tyagi.
ठीक है, मत बोलो, रहने दो फिर.
Excuse me. So some of the like bulbs you can make in garlic. Dr. Ruchira has developed a protocol where in vitro you can have the bulbs, and these bulbs can be stored, remain in tissue culture tube for longer period of time. And the advantage was that you can take this bulb after, you don't need to subculture, you need to take this bulb and can distribute to the farmer, take to the field, or you can multiply material from there. And similarly, microrhizome was developed, the protocol was developed by Dr. Tyagi in ginger, where you can have a small rhizomes in it. And these rhizomes can be kept even at the 25 degrees in the room temperature, and they do not, you can use it for distribution, or you can use it for multiplication. These were very good experiments. And microtubers can be induced in taro, I guess. This is taro, where you can have the microtubers. These increase conservation period, as I said, that in other crops you need to subculture. When you see the media browning, then you need to subculture it. But storage organs remain there for a longer period of time. Growth retardant, temperature, mineral composition, medium, photoperiod are important factors that promote the storage organ. Normally, I've seen that increased sucrose concentration give rise to the microrhizomes and bulblets. The storage organs have been successfully induced in vitro in species of tulip, yam, potato, and ginger. So this is one way of in vitro conservation. Then we move to long-term conservation. Whatever I was telling was short to medium-term conservation, which is happening in the in vitro gene bank. And for cryopreservation, which is a long-term conservation, where we take the explant and develop a protocol, put that explant into the liquid nitrogen, and the germplasm is stored at 196 degrees centigrade. And it is said that theoretically all the activities, metabolic activities of the material, it reduces, thereby the conservation goes up to the infinite. Several explants can be cryopreserved, and here we are talking about in vitro conservation, then the shoot tip and the nodal, mostly the shoot tip or axillary bud we prefer, but sometimes the meristematic clump, like in banana, that protocol was established, and that can be used also. And the somatic embryos, if a crop is there where somatic embryos are there, they can also be cryopreserved using different techniques. Protocols need to be optimized for individual species and sometimes individual genotypes. So these protocols are crop specific, and if one protocol is established for one species, you can try for different accessions of those species, and little bit changes may be sometimes required. And during the development and or improvement of the protocol, both cryogenic and non-cryogenic factors need to be carefully balanced. So when we develop a protocol, we see that without freezing, how our explant and how much survival and how much regrowth we are getting. And then after the cryopreservation or freezing in 96 degrees centigrade, how much plant is growing. So once we have developed this protocol using different techniques, I will show you in other slides that what all techniques are used for that, and you can choose techniques according to your lab conditions and the expertise, and you can cryopreserve that material. So our emphasis right now is moving those whatever we have in vitro to move them into the cryo gene bank for long-term conservation. But the protocol development is really takes a lot of time and a lot of efforts and a lot of training. So there are two types of cryopreservation techniques. One are said as classical techniques, and the others are vitrification based techniques. So this one is control rate freezing, which is a classical technique, and it is used for dormant buds. In USA, they have conserved about 2,000 accessions of apple using this technique, where they have collected the samples and then explant was isolated. They have used for 1 inch old, 1 inch long vegetative tissue with the one axillary bud, and put them in vials like this, polyolefin tubes. And then they went for programmable freezer. In this control rate freezing, you need a programmable freezer where to set the program, and the temperature is reduced slowly, and later those explants will be, and the moisture content reduces. And here again, this is a USDA Fort Collins cryobank, where they have several of these kind of tanks and put them in liquid nitrogen. And after liquid nitrogen treatment, you see the viability, if the viability, how much viability is there. So the method for knowing the viability is tissue TTC test. And after that, these buds directly, they graft them on the root stocks, or after grafting, it grows. So after that, they can see how much regrowth or the establishment of vegetative bud on the root stock is. So here you can see this flowering also. This protocol is from USDA and for apple. The other technique is encapsulation dehydration and encapsulation vitrification, where you have a plant sample and desiccate those. In encapsulation, you have to make a bead kind of thing. And in other lecture, which is a practical, probably you would know much more about these techniques. Here, the shoot tips are excised and they are encapsulated into the sodium alginate, and then they are desiccated under the laminar flow and then put into the cryotube. And before that, they are also treated in 0.75 or 0.1 molar sucrose for overnight osmotic desiccation, and then air dehydration, and put them in liquid nitrogen. And after liquid nitrogen, thawing, thawing meaning rewarming, which can be done under, for this encapsulation dehydration technique, can be done under the laminar flow. You take out the beads and keep it at the room temperature, that is slow thawing, or you can do it under the water, 37 degrees centigrade water or 42 degrees centigrade of water. And in 2 steps, you can give those cryovials into that water, and then you can take them out and put them in the medium for regeneration. And other is encapsulation vitrification, where after encapsulation, the material is the beads material which is encapsulated, and we are calling them in encapsulated beads. They are treated with PVS2 solution. So the osmotic desiccation is done in PVS2, and then we are putting them in liquid nitrogen. Here the thawing is again, here slow thawing is not done. We are doing the fast thawing in the water. And the plant is ready for after cryopreservation. We put them in regrowth media. This regrowth media is also special for species. So it is also being standardized. So the multiplication media is being standardized, and the regrowth media is also very crucial for giving you good survival and regrowth after cryopreservation. The techniques are vitrification based techniques, where the plant is there and to take the shoot tip out. This is basic. This is a technique where you use vitrification. Vitrification is the method where the solute of the cell, they are vitrified into the glass. So the ice does not crystallize into the cell. So when ice does not crystallize, it means the cell remain viable and cell wall doesn't break, and it gives you the good growth. So there are several different steps and different techniques also, like vitrification, droplet vitrification, decryo method, V-cryo method, and cryo mesh method. So these are little modification. Vitrification uses explant, and then you can put them in PVS2 or PVS3 and directly put them in liquid nitrogen, and after rewarming, regrow them in the medium. For decryo, the decryo and V-cryo was the technique which was developed by Takai Rino from Japan and Yamamoto and their group. And this technique is there they have made a small cryoplate kind of thing, and which has wells in it. So your shoot tip and explant is put on the cryoplate, and with the help of alginate and calcium chloride, these are sticking onto the cryoplate. And then this in decryoplate, these are directly dehydrated after loading solution treatment. They are dehydrated under the laminar flow and put them in liquid nitrogen. In the V-cryoplate method, PVS2 treatment is given, and after PVS2 treatment, loading solution and PVS2 treatment, they are put directly into the liquid nitrogen. The advantage of this technique is that the shoot tips are stick onto the cryoplate, and you can use 10 or 12 shoot tips at one point simultaneously. as in encapsulation dehydration, you have to use single bead. So it's a lot of little laborious. In vitrification, you are handling direct meristems. So sometimes we lose those material meristems because they are very teeny, very small, and we can lose them with we lost them. We lose them because sometimes they are coming with the in the pipette and drop somewhere here and there. The other technique, which is droplet vitrification technique, where we are using aluminum strip and put our material into that and directly put PBS2 into that. So there also the material or explants are clumped together on the aluminum strip, and we can directly put it in liquid natural. In banana, it has been this technique has been very efficiently used, and this droplet vitrification is very, I mean, it gives a result in different species, which has been reported and very successful. And this cryoplate and weak cryoplate, only thing that we generally it is very difficult to obtain this cryoplate. But this technique is also good because the loss of explant remains almost nil. So here are some of the experiments which were done. Diascoria deltoidea, again by Dr. Neelam Sharma. So we can see the cryopreserved and non-cryopreserved material, and these plants have been put in tissue culture and then in the field. And in Gentiana, again, the Neelam madam work we are showing, you can see the Gentiana is a medicinal plant where you can see the growth after cryopreservation, and these plants are in the tubes. This work was done in our lab on strawberry, where you can see after liquid nitrogen treatment how much growth you can get. So this is, and without callus, so these cultures are growing and then transferred into the tube and growing very well. So these are these explants were cryopreserved using weak cryoplate method. And here you can see the vaccinium, which is a blueberry, and the encapsulation. These kind of beads become the encapsulated, the shoot tip. We have taken the shoot tip, and which was encapsulated here, and air desiccation and osmotic desiccation, and then here you can see after cryopreservation, the new shoot is coming. So now I would like to tell you about our in vitro gene bank and the base gene bank, in vitro active gene bank, where the cultures are kept. Like you can see the test tubes, and this is maintained at the 25 plus minus 2 degrees centigrade. And for long-term conservation, these cultures, we take out the shoot tips and process them using any of those techniques which I have just elaborated, and put them in liquid nitrogen, like these kind of boxes. These have cryo vials in it, and then our processed material into it and cryopreserve into the liquid nitrogen. So for long-term conservation. So these are little comparison we have done for in vitro active gene bank and in vitro base gene bank. It is active collection and it is a base collection. And then this is for short and medium term storage, and in vitro base gene bank is for long term storage. And here the cyclic flow, we are subculturing the material after a period of time when the shelf life of culture or culture growth is not good enough, and then we subculture. So one cycle is being followed again and again for a particular species. And here, once we put this material into the in vitro based gene bank, we do not disturb it. Normally, we don't distribute. For distribution, we are using this active collection. And for base collection is for future use. In case of emergency, you can take the material out of the cryobank, but we prefer not to open the liquid nitrogen tanks and distribute the material. And here it is controlled culture condition. The plant is stored, and in vitro gene bank liquid nitrogen, as I said. And in storage temperature here 49 to up to 15 degrees centigrade, but here 196 degrees centigrade. And in vitro active slow growing of shoot, but here the these are the explant, but here you can have shoot tips, zygotic, somatic embryos, cell, and protoplast, depending on your objective, that what species you are working and what is your objective. If your objective is in vitro conservation, then especially we are doing the shoot tip, or but zygotic and somatic embryos can also be cryopreserved. Metabolic activity slows down over here. The germplasm is suspended growth, so metabolic activity is stopped. This slide shows the status of in vitro gene bank at ICR and VPGR, where we have about 1,966 accessions belonging to different crop groups. And we have 63 species, and genera is 156 species, about 39,000 cultures, and these are being periodically subcultured and maintained at the culture room temperature. And these crop groups are being handled by very special people who are specialized, who are dealing with by scientists. One crop group is being done by one scientist. So these scientists have developed their expertise to handle these crops, develop protocol, a lot of work, develop protocol for from micropropagation to up to in vitro conservation, acclimatization. And then we also do the genetic stability of germplasm, because to avoid or to ensure that the germplasm which we are conserving is having its or maintaining its genetic integrity. And this slide shows in our collection in vitro based gene bank. So we have these kind of tanks which are kept here, and these kind of the boxes we have, and in these the cryo vials are being arranged. By now we have 298 accessions stored in, and about 136 accessions of allium. And this has major contribution of Dr. Ruchira Pandey, who has done work on this, and Dr. Radha has worked on Musa species and cryopreserved a lot of material. And you can see the techniques which are vitrification, droplet vitrification, and encapsulation dehydration, and the explants are shoot tips, basically, mostly the shoot tips. Basically the shoot tips, mostly the shoot tips we are cryopreserving. And here you can see our cryo gene bank, where we have these big cryo tanks. And the explant herein are some of the orthodox species, orthodox seeds are also cryopreserved of those varieties which are very important, rare endangered species, where the material is less. And as you know now that for gene bank, for cross-pollinated crops, there are a requirement of 4,000 accessions, and so 4,000 and 2,000 seeds are required to conserve in the seed gene bank. But here, if the material is less, say about 100 seeds and all, we don't throw it away. One set is sent for multiplication so that the germplasm can be multiplied, and the seeds can be stored in seed gene bank. But if quantity is less, then it is given to us for cryopreserve. And then the procedure is to reduce the moisture content and put the seeds in the liquid nitrogen. But here also the tissue culture technique comes into the play, where the embryonic axis of the seeds are taken out and cryopreserved. Then the germination of embryonic axis is done in tissue culture media. So many species like jackfruit, like lychee, the protocols have been developed for recalcitrant seeds. And otherwise, we are doing gene pool conservation here, because a lot of germplasm is available in the field, like apricot, walnut. We have a lot of germplasm, indigenous germplasm, which needs to be cryopreserved. So we have the material in the field gene bank, but the duplicate set is here in cryogene bank for future use, or for posterity, you can say. This slide shows the status of cryogene bank, where we have different crop groups again, and also we have dormant birds of mulberry and other crops, and then pollen grains also. Pollen grains are very important to cryopreserve. Like in mango, mango is recalcitrant for tissue culture. It does not grow in tissue culture, though the vegetatively propagated crop, but seed are very big. We all know about mango seed and all. Then the pollen grains of mangoes were cryopreserved and were used also for breeding purposes. These were shipped to, much of the pollen grains came from Lucknow, the subtropical institute, and then those cryopreserved mango pollen were taken to the field in Lucknow and were used for breeding, and those plants which were used, where the cryopreserved pollen were used, they could bear the fruits. So it is a very good way of conserving the germplasm, where other methods of conserving is not available. Then for disease eradication, I have shown you in the first slide that tissue culture is also used for disease eradication, especially for virus infected plants can be treated, and we can get virus-free explants. So the steps are that we need to know that these are the infected plants from the virus. Raise the cultures, shoot tips, excise the shoot tip, and the good part is that liquid nitrogen has played a very good role to give the virus-free cultures, and it has been in literature, many crops. This cryotherapy has been used. Otherwise, meristem culture has been used for several years for getting the virus-free material. But now, very recently, cryotherapy has been used to get a virus-free material. And after cryotherapy, post-culture for plant regeneration. And then again, the plant which are regenerated after cryotherapy do the virus indexing. Then once the cultures which you are getting is through that there is no virus, then we can have the virus-free stock in plants. Any technique of cryopreservation can be used for this cryotherapy. This one slide, I would like to just show you that how cryotherapy works. So here is a shoot tip. This work has been done by Wong et al. And this is on sweet potato. So the shoot tip, you can see, and they have used encapsulation dehydration or encapsulation vitrification. There you can see the shoot tip is enclosed into the encapsulated into the bead. This bead is of calcium alginate and processed. So after cryotherapy, so first of all, you see the virus infected plants. What does it have? It has virus infected cells. It has healthy cells. Then it has again healthy cells and virus infected cells. These are the leaf primordia. This is apical dome. So this is what is the shoot tip. And then once you do the cryotherapy, you can see these are the killed cells and these are the healthy cells. These are still some of the cells are surviving virus cells, but most of the plant cells are virus infected cells are killed. So when this kind of case comes, we need virus-free plants. So they suggested that once we give the thermotherapy to the plant, and then we can go for cryotherapy. So when this thermotherapy was given to the shoot tip, then virus infected cells, they are there. Then healthy cells and virus infected cells, they are still there. And then after cryotherapy, you can see all the virus infected cells which were there and they are killed. So that is how we get the virus free material. And the advantage of this is that you have a protocol and you don't have to have an extra set of equipment or anything. You can just go for cryopreservation and cryotherapy and you can have the virus-free material. And sometimes, direct cryotherapy can also give you the virus, like this stage can also get, depending on the that how much infection is there in the tissue. Then tissue culture is also used for, pre-breeding, and where you can have the hybridization program, where you have the embryo which are immature and abort and those kind of problem you are having. Then you can have different techniques like protocol culture and microscope. Microscope can be used, ovary and ovule culture and embryo rescue culture for the use of these programs. And here you can see the selection of how the embryo rescue is being done, selection of immature seed, then sterilize it, and sterilize seeds, you take out the embryo, so embryo, excision of embryo, and regenerate on the media, then develop a plant, then seed, and you can transplant it and get the new plant. So this kind of thing are these. We are also using in tissue culture unit from Bigna and other crops. Embryo rescue technique has been used. So in conclusion, I would like to say that in vitro techniques, they offer invaluable complementary approach to secure the diversity in PGR. Yes, you can have the vegetatively propagated crops in the field, but you can have the duplicate set in recently it happens like in Bhowali, the landslide happens and the Fragaria accessions were largely damaged. And we have some of those accessions duplicated in our in vitro gene bank in a tissue culture form, and we suggested that we can replace those cultures, those accessions to the field again. And the techniques aid in managing germplasm to ensure loss of valuable genetic resources. And a wide range of techniques and protocols are available for use. It depends on, the resources, the technician, what, and also for the genotype, that what crop you are being handled and what kind of, how big your gene bank, in vitro gene bank is. So you can choose any technique which is, suitable to your species or plant, or suitable to your lab. And you can choose any technique and you can go for the in vitro conservation. This will be for active collection, and then you can slowly move these collections to the cryopreservation. And then you can also, after you put this in vitro collection into the cryo banking, then you can, free to work on new species and new genera and develop a protocol. So at the end, I would like to thank organizers, director and BPGR, that giving me opportunity for, giving this lecture. And I thank the staff of TCCU who really work throughout many years put their efforts to develop protocols and conserving those germplasm starting from the 1986 and up to now, doing efforts towards the conserving the germplasm. And I would like to thank Dr. Ira Malhotra, who is scientist in TCCU, who has really helped me to have this presentation on the write-up. And thank you very much.
Thanks so much, ma'am, for a nice presentation. And now I would like to request our participants, if they have any question, please ask. Ma'am is here to answer the questions related to if you are working in any specific species or any specific area of tissue culture, then you can rightly get the guidance from ma'am.
Hello.
Yes.
Hello, madam.
Yes.
Good morning, madam.
Good
Good morning,
morning.
madam.
Good morning. Good morning.
Thank you, madam, for a wonderful presentation in your side.
Thank
Myself,
you.
Dr. K. Gupta from JNK Jabalpur. Madam, I know I asked one question. In tissue culture, in fact, virus infection plant, if you go to some tissue culture, raise some tissue culture plant, so how many years survive for tissue culture material?
If you keep on subculturing, it will survive for many years. You don't let the plant material die, but
Right.
keep on subculturing and it will survive for many years. This is how we are having our in vitro gene bank running through regular subculturing for say about many years,
they generate
20,
many
30 years. Yes.
Thank you, madam. Thank you very much.
Yes. Thank you. Any other
Any other-
question?
Any other questions from participants? Today,
I think everything was very clear.
I am.
Or people are not interested in tissue culture, maybe. They're not
No,
working.
no. It was a nice presentations and everything was very clear.
Thank you.
So if there is no questions, then I would like to thank Sandhya ma'am for giving us a nice presentation and highlighting importance of tissue culture and how tissue culture and cryopreservation can really be beneficial, especially for saving some of the very rare germplasm. And also I want to say my participants that if they are working in a specific species, because ma'am is handling so many species in her lab, so many scientists are there. So in future, if they want to work or are facing any kind of difficulty, they can interact and get in contact with ma'am and the lab mates there. So I think they will all be happy to offer help to them. So with this, ma'am, I would like to again thank you for
Thank you,
being
madam.
with us.
Thank you. Thank
Thank
you.
you so much, ma'am.
Thank you.
Okay, so now we have a gap of around 1.5 hours, sir. Because our next lecture will start with Dr. Ashok Kumar, sir, and it will start from 12:15. So we will be back by 12:15 to start our new session. So I request all the participants to please be available timely. Is it okay, sir? Huda, sir? Sir, you are muted. Sir, you are muted, sir.
12:15.
12:15, sir, before lunch.
Okay, okay. ये पहले वाले में नहीं हो सकता था अभी?
No, sir, actually, sir has
11 बजे?
requested, no? Yeah, sir requested, so the
Okay,
timing was
one and
adjusted.
yeah. Okay.
Yeah. And after lunch, we have one practical session. on tissue culture, means cryopreservation and in vitro conservation. Is it okay, Subhash sir?
It's okay, madam. And Dr. Ashok sir का लेक्चर है ना अभी 12:15.
हाँ 12:15 से है.
हाँ ठीक है मैडम.
ओके सर.
चलो ठीक है मैडम, 12:15 बजे मिलते हैं फिर ना.
ओके चलिए.
ओके.
Okay. Okay, so I'm starting. So now our another speaker for today's session is Dr. Ashok Kumar sir. So I'm very pleased to introduce Dr. Ashok Kumar sir. So we have his brief biodata here. I will share my screen to share it with you all. Our speaker, Dr. Ashok Kumar sir, he is currently principal scientist in the discipline of plant breeding at Division of Germplasm Evaluation at NBPGR. And before this, he has held many renowned posts as well. He had joined as a scientist and continued as till 1996 as senior scientist, senior scale, then the senior scientist from 1998 to 2006, principal scientist as till 2014, then principal scientist and head for 2 times from 2014 to 15 and 17 to 21. And he also served as the acting director of NBPGR from 2021 to October 2022. And his nature of research work includes the duties related to management of plant genetic resources with special emphasis on characterization and evaluation. He is also the program leader of characterization and evaluation of agri-horticultural crops. Then he is also the coordinator for the lead center on agrobiodiversity management and utilization of agrobiodiversity in network mode. He is also the coordinator of one of the mega projects being conducted at NBPGR in Linseed, and also the coordinator of other 2 mega projects on sesame and chickpea. And he is also involved in the large scale characterization, evaluation, and development of core, mini core, and trait specific reference sets in different crops like wheat, barley, lentil, many other minor crops as well. Then he has identified and registered more than 50 unique and trait-specific germplasm in different crops. Then he has also served as an expert reviewer for the development of the descriptors for many of the crops for their characterization, evaluation, and utilization. He has many publications, more than 110 research articles in national and international journals, one book chapters more than 20, then technical and popular articles and many more. and he has been also recognized as fellow of Indian Society of Plant Genetic Resources and fellow of Indian Society of Genetics and Plant Breeding. So with this brief biodata, I will stop sharing my screen and request Dr. Ashok Kumar to please continue the session with his lecture, sir. So, yes, sir.
Thank you, Dr. Mamta. Good afternoon. I am really thankful to our young colleagues, Dr. Mamta, Dr. Jyoti, Ravi, or Kuldeep, for giving me opportunity here to interact with all of you and to share my views about the descriptors, descriptor states, and the development of the descriptors. So I am also thankful to our trainees. It is visible.
No, sir, still it has not started.
No.
Yeah, it has started being shared.
Yes.
Yes, sir.
Sir, it is not in presentation mode, sir.
Is it all right?
Sir,
Not
make it
it,
in
sir.
presentation mode, sir. In slide mode, sir.
ये हैं अशोक कुमार
अरे सब सही
वाले थे है ना
It is coming, sir.
Is it all right?
Yes, sir. It is all.
Sorry for delay. So really I am thankful to all of you to be here this afternoon and to discuss about this descriptor development and the recording. So then why descriptors are needed? Actually, we have got so many exceptions in the gene bank. These exceptions are not being characterized, then the gene bank and to be considered as a black boxes. So just to utilize the gene bank or the gene bank material, this is quite necessary to characterize, to evaluate our material. So here you can see the utilization of the germplasm, gene bank collection, and the characterization evaluation, then effective germplasm management, then sharing of the germplasm for utilization, then the development of the product in the form of variety, in the form of the genetic stock. So in whole the series, we are required the traits or the descriptors here I will talk only about characterization, and evaluation. So then what are the descriptors? Descriptors are the characteristics, or you can say the morphological traits used to describe a phenotype, to distinguish an accession. Here, traits can be scored or measured and expressed in the form of numeric value or in the form of the qualitative traits. So then the set of descriptors, or you can say the set descriptor used to facilitate the data collection, data classification, storage, and exchange of the data and exchange of the journal plot. When the descriptors are at one place, then we make the descriptor list, the set of individual descriptors to describe an accession, to distinguish an accession based on the number of descriptors. So you can say the number of descriptors become the descriptor list. And the descriptor states the state of the expression is clearly defined in characteristic to harmonize the descriptor a particular stage, or it represents the variation, observation. So for that, you have to require a lot of variation in our population, in our germplasm, just to identify the descriptor as well as the descriptor states. Here example has been given about the stem pubescence. The descriptor is the stem pubescence. What kind of pubescence? So here the descriptor states the sparse, the dense, or intermediate. So descriptor and descriptor states both are very, very important just for the meaningful characterization as well as for the evaluation of the germ plot. Then why we need the descriptors? That these are highly required for the characterization of the material, the standardization of the information, and the establishment of the databases for the utilization by the breeders, by the crop curators, by the germplasm curators, and the documentation of the germplasm, documentation of the accessions in our gene bank, and creation of, just for the management of, based on the descriptors, management of our large number of accessions, creation of core collections, core set based on the morphological data or the descriptors, and overall the exchange of the data among the other gene banks and among the other the persons who are going to utilize the germplasm. Then what are the descriptors? Crop descriptors. These are the different categories of the descriptors. The passport descriptors. These kind of descriptors are to be collected by the plant explorer on site itself. For example, that includes the plant data as well as the site of collection, the site data. Then we come to the management descriptors that to be required for the management of the gene bank. So such kind of descriptors like germination or viability test. So there are so many descriptors for management of the accessions in gene bank, and the site and environment data for the characterization, even for the exploration. The site data and environmental data are quite important. So the environmental descriptors, site descriptors are to be recorded specific to the plant data at the time of exploration or at the time of the collection and the characterization evaluation data. So we are very much concerned for the utilization of the germplasm. So for the characterization, are to be required. It depends upon the crop to crop. It varies from crop to crop, species to species. And then lastly, the evaluation. Evaluation descriptors are to be developed by the experts along with the breeders, the trade, what we are going to evaluate the germplasm, etc. So the rigorous evolution discussion is to be developed with the help of the crop curators as well as the breeders and the actual the persons who are going to evaluate the screen the germplasm, evaluate the germplasm. The characterization descriptors, just this is the initial stage of the germplasm characterization to identify exception, specific trait to make selection for further research in breeding and to identify the group of the morphologically, genetically similar or dissimilar groups. So how we can record such kind of information? The observation about the plant characters used for the just to distinguish the accession or to describe a phenotypic variation, and in the characterization descriptors, the traits should be highly heritable and it should be expressed in all kind of the environment for the characterization of the because we are using characterization descriptor just to distinguish an accession to the others. So in any condition, in any environment, it should be expressed. It should have it has no the G into interaction or the low G into environment interaction, and the characters scored visual in the morphologically the characters or the traits recorded for characterization as characterization descriptor should be visible and highly distinguishable and to be seen by the naked eye, and the characterization descriptors should be discriminated by the accession variation in the accessions, and these characterization descriptors should be controlled by all major genes, or you can say the highly heritable or the major genes. Then what are the limitations of the germplasm characterization? Mostly the qualitative nature only gives an idea of the extent of variability. For example, it will help in the categorization of the germplasm, large scale characterization help to in the grouping of the germplasm. So such kind of characterization descriptor or the distinguishable traits can be taken into the potted experiment. So even sometimes not any kind of design is required, just to check to see the visual characters in particular genotype. And the initial sample collected the heterogeneous and will not expect any conclusive idea about the informity of the accession. So here the homogeneity is required for the characterization of the descriptors, particularly. So these are some of the limitations for the characterization of the germplasm. Then we go for the evaluation descriptor. Now the evaluation descriptor is very, very important for the utilization of the germplasm. So now the great interest have been given, imperative have been given to the curators with the help of plant breeders, because such kind of descriptors or traits are required for the crop improvement program and for the domestication of the new crop. These include some yield parameters, agronomic traits, biochemical traits, the screening or the traits related to the biotic and biotic stresses. And the evaluation descriptors should be undertaken in a very thoroughly, particularly in appropriate experimental design, statistical design for the use of the data. And sometimes it needs the replicated trials, replicated data, replicated trials in the form of check or in the form of the treatment, just to minimize the error and to identify a potential germplasm for utilization. Evaluation descriptors are very, very important and to be taken with due care with due care, with the help of not only the crop curator, breeder, and along with the experts of this, you can say this entomologist, pathologist, or the physiologist for abiotic stresses. Then the descriptor list a set of individual descriptors used for the description of the germplasm. So in gene bank, accessioning, description of the germplasm, description of the germ,
किधर है मंदिर?
first of all, the descriptors, with help of descriptors, we go for accessioning based on the descriptors collected in by the explorer.
Please mute yourself. All are requested to mute yourself. सर आपकी आवाज नहीं आ रही सर. सर अनम्यूट कर लीजिए. सर आपकी आवाज नहीं आ रही है.
It's all right.
हाँ सर ठीक है.
So first of all, the registration in the gene bank. Here, registration in the gene bank means the accessioning of the germplasm based on the descriptors taken or collected in the field, collected at the time of exploration. Then the descriptors are required for the characterization and evaluation. Then the management descriptors for in the gene bank, and overall the characterization and evaluation descriptors are the key attributes for overall the utilization of the germplasm. So here you can see the descriptor list includes the key attributes or the characters or the traits of a crop and set out method used to measure and document them along with the relevant accessioning data. Now we come to the minimal descriptor, because the descriptors are too exhaustive. So as a germplasm curator, as a plant breeder, we have to develop our own descriptors for the germplasm purposes. So the descriptor developers can make out minimum set of highly discriminating characteristics of the descriptor to record in each accession. To the restricting recording will save the time and labor of the characterization. Here we have to take the limited number of the characterization descriptor or visual descriptor just to save the time and labor, and to the minimal descriptor will help us to indicate the diversity in the collection, and it will provide the harmony in the documentation system and the exchange of the data among others. When after defining the descriptor, the range of criteria that can be applied for most of the practical descriptors: the type of data, the type of scale, the resolution, complexity and cost, method of collecting data, experimental design, scoring, coding, numerical, and alphanumeric coding scheme. So these are the data, and these are the scales we have to go for the descriptors for analyzing and utilization. For the qualitative data, the limited number of states, descriptive states, so the limited number of the should be used to define a descriptor state, and list of the distinct descriptor states must be very much distinct. For example, I have given the example of the seed coat texture. So descriptor stays smooth, partially rough, radially rough, partially radially rough, and reticulately rough. So these are the, so before going to the such type of information or taking the descriptor into account, we must thoroughly see the descriptor. For the descriptor states okay. Then for quantitative data, because evaluation descriptors need quantitative data, so it will cover full range of variation in the germ plant and the different states of expression recorded using discrete data. For example, the days to flowering and the continuous variation that are measurable in case of plant height or the you can see the continuous trace or the polygenic trace particularly that are being utilized by the breeders or you can see the breeder driven trace as a evaluation descriptors. Now we come to the descriptor method. First of all, an object. The object of this descriptor method, the plant, exact part of the plant. If you are taking the root into account or the stem into account, the all measurement should be given into the SI system. So similarly, the condition, you can see the state, the condition under which the observation have made the full expression. You can say that the growth plant growth have all the phenological conditions at which the descriptor to be recorded. And the sampling procedure, that is very, very important for the evaluation purposes, the number of samples on which the observation is to be recorded. So the accuracy and the full range of variation present in the population should be described. So the sampling method, for example, in case of self-pollinated crop, it around 10, for example, 10 plants, or in case of the you can say in case of the cross-pollinated or the allogamous crop, it must be around 20 to 50 plants per accession or per sample. So these are some basic rules for the descriptor development. That we have to keep the descriptors as simple as possible and can be used for wide range of the users. Similarly, the drawing and images to support the descriptor clearly to clarify the complex descriptor and provide clear definitions of the descriptor. So the descriptor should not be the ambiguous; it should be unambiguous and analyze critically the unit cost of the measurement in terms of both. In terms of the labor, in terms of the material, in terms of the even in the terms of the time, because the time is very, very important. It will if it can use more time, then the stage will go on for the to record descriptor. Then consult widely crop and gene bank specialists to achieve comprehensive and understandable list of descriptors for wide acceptance because the data to be used by other stakeholders. So all kind of stakeholders must be aware of the recording of the descriptor. So the keep the descriptor as simple as possible. I've already told. Then use the
Please check your mic is switched off. It's creating noise and disturbing in between.
So they analyze carefully unit cost per management and measurement. So just to see the cost for characterization and recording the descriptors. I have already told them about the ambiguity.
शाबाशी बोलो दे
Before going to the development of the descriptors, first of all, we have to investigate the range of diversity for the trait in if no diversity is known, so descriptor is necessary. The descriptor name should be descriptive, unambiguous, and compact. Avoid developing descriptor that describe more than one character at one time. For example, the leaf blade color. So the white and stripe, pink and mottled. So you can here you can see you can take two descriptors, pink and the other is mottled. We cannot mix the descriptors and better to split the descriptors. The leaf blade color, the leaf blade variegation like this, mottled is the variegation, and this is the pink is the leaf blade color. Similarly, define how to trace should be recorded, visual assessment. So for that, we have to take a lot of germplasm population to deal with the development of descriptor. So list the distinct descriptor states you want to use. Use continuous scale, particularly to determine the labor of the resolution needed when the possible include the actual value of the measurement. Using this the SI units. Similarly, define the descriptor state. When at which state you want to take the number of these starting from 1 to 9 scale. For example, the states are for flower color states are red to white, pink, purple, or the violet. These are so many states. So that to be categorized as a scale 1 to 9. Then the reference is made to a specific method, system, or provided relevant biographical references, give a full citation, cultivar, reference variety, germplasm available is widely known. Descriptor the descriptor should be highly discriminatory. If the descriptor is discriminatory, then you mark, and this will indicate the descriptor particularly useful and indicator of diversity in the collection. But whenever you come to across a new trait or new descriptor, so new stage descriptor stage you can mark as a 99 in that case. Then we come to for recording quantitative traits. So I have already told in case of pollinated crop 5 to 10, and cross-pollinated crop of 10 to 25 plants to be taken to take the average, to take the population taken into the account. For most of the qualitative characters, if the descriptor states is not in our descriptor state, then you can specify that is others, or you can, as a remark, you come across any new states, then you can add as a 99 or add others as a descriptor. If particular descriptor is inapplicable, then this kind of descriptor we mentioned in the inapplicable, and with the notes as the under remark that the descriptor is not applicable. So when we go for the descriptor development, so the germplasm or the population should be free from any kind of infection or infestation. just to see this really the phenotypic phenotypic trait, or it has been due to this disease or some other infection. So similarly, so the date should be expressed in the numeric format given here. Similarly, to record this color, so the standardized color should be utilized, the Royal Horticulture Society color chart, to just for the to know the ambiguity of the states. Just for the RHS chart are useful, or they have been accepted at universal level, just to identify the particular states, and to the it will help in exchange of the information particular state. Then the descriptors should be as simple as possible, and it should be understood by a layman, and can easily be used by a range of the users. So there are so many agencies are developing the descriptors, the biodiversity, the IBPGR. These are the dealing with the germplasm global level, including the CG Center. They are developing descriptors, thorough descriptors for the characterization descriptor, for the evaluation descriptor. And the NVPGR earlier have been using the descriptors developed by the IBPGR, but during 2000, the minimal descriptors for the characterization and evaluation, preliminary evaluation of the agricultural crop was developed by the NVPGR scientists in collaboration with the crop experts or the crop advisory committee. So NVPGR has developed the descriptors for the field crop, descriptors for the agricultural crops, medicinal aromatic plants, fruit crop, vegetable crop, and there are so many are in the pipeline. Then some of the other national organizations like PPWFRA are also developed the descriptor for the distribution variety for different crops. Then some of the CG centers like IITA, CARDA, they are also developing their own descriptors for characterization evaluation. And recently in 2010, the Gracia had developed, in collaboration with the crop expert, they developed the utilization descriptor, other than this characterization evaluation, the utilization descriptor. Then we come to the how discovery we have. This is the case study I have taken in 2 crops, and my colleagues, Dr. Kuldeep Tripathi and other at the Choose Center, they have developed the descriptors for new crop, the Vigna vexillata. So how they have developed the descriptors? So first of all, they have augmented the germplasm from different sources, and they have seen the variability. They have minimized their the descriptor, and over the years of over the years of the you can see this growing the crop, they have come across the 56 distinguished descriptors comprising 37 are the qualitative and 19 are the quantitative in case of the Vigna vexillata, the new crop. So these are the, for example, these are the descriptors like seed germination, this is the stage, and this is the observation. So here I have taken this one as a, you can say, as a leaf character. First of all, the plant character, the leaf character, the stem character. These are the glabrous. So here you can see the descriptor states as well as the stage of the recording of the information or the data. This is the thorough study of this the tuber cowpea. So these are the some of the traits. They have been descriptors, the quantitative descriptors as well as the qualitative descriptors have been identified in this new crop. Then this is the leaf shape. This is the example of one of the descriptor in case of the tuber cowpea. You can see the drawing of this the leaf lanceolate, elliptic ovate, the off-lanceolate ovate, elliptic. So here you can see these are some of the botanical descriptors, because in case of the crop native or CWR or new crop, we have to be more cautious to record this botanical descriptors, because these are very important, because during the evolutionary study, this such kind of the descriptors will help to delineate the species or to see the variation based on the agrobotanical descriptor. Then similarly, our tissue centers, Dr. John and Dr. Sumar and Pradeep, they have identified device descriptor for this Momordica Cherencia muricata. So I have already told that for taking consideration into the fruit color or flower color, we have to take the RHS color chart into consideration, because based on this number, we have to see the exact color or object color to further the acceptability global level using this RHS color chart. Then similarly the fruit shape, the cylindrical, the spindle shape, or the glow pose. So here you can see the variation in the germ plant. So our purpose is to say here the germ plant variation is the variability in the germ plant is very much required to describe a descriptor, to divide a descriptor. It may be the agrobotanical traits, or the qualitative traits, or the traits of this quantitative. Then you can see the variation in the fruit of this Momordica, oblong, discoid, rhomoid. So this is, and similarly in the seeds, you can see the variation in the seed shape, seed surface, seed ornamentation. So here you can see the rectangular, the first one, then narrow rectangular, then square. Then so the variation is required. The variation is required in the germ plant to divide the descriptor or to divide the descriptor states. Then some of the descriptors have been developed in new crop like Jatropha and this Momordica, I've already told. Then the Vigna vagulata. So this is very important and very difficult to develop descriptors, particularly in the new crop, the underutilized crop, the neglected crop, or the crops are not in the public domain, because we have to see the traits, we have to see the characters, we have to identify the state, and we have also divided the stage of recording the information. So the development of descriptors will not help only in the characterization or the evaluation, utilization of the germ plant. It is also helping to popularize underutilized crops, neglected crops, or the crops of the the lesser known crops, based on the characterization data, based on the descriptors. It is it may be the agrobotanical, it may be the qualitative or the quantitative. It will help to utilize the germplasm and to popularize germplasm or popularize new crop. With this, I would like to add one more thing. In case of the established crop, for example, wheat, rice, maize, now we are also in the process of the development of the utilization descriptors, evaluation descriptors, and the utilization descriptors for the thoroughly discussed with the help of the crop expert, with the help of the experts in the particular field, maybe the biotic stress, abiotic stress, or the quality parameters. So these are very, very important for the utilization of the germplasm. With this, I am thankful to all of you for giving me opportunity to say few words about the descriptors. And I am thankful to my colleagues to giving me opportunity here to be interact with all of you. Thank you.
Thank you so much, sir, for a very nice presentation. And your lecture was really very important for this training on PGR management, because descriptors are the basic things that will explain the our germplasm. And also you explained the role of NBPGR in devising descriptors in various crops. So now I would like to request our participants, if they have any questions, they can ask sir, and he will answer the questions. One of the questions that I can see in the chat box, sir, is how much, it is from Dr. Shashikant Patel, and he is asking how much variability is necessary for development of descriptors.
How many? Validity.
How many variability is needed, but to what extent this variability should be for the development of descriptors in particular crop?
For crop by relative or the new species, so the variability is required, particularly for the development of the descriptors, because if the variability is not there, then it is difficult to devise a new descriptor. So variability is required. In case of the established crop, the quantitative variability is required, but in case of the new crop, the qualitative variability is required, or the botanical variability is required, particularly for the identifying the new descriptor or new traits or new information in case of new species.
I hope that he got his answer. So another question is from Dr. Hridayesh Anuragi. He is asking, for trees, what will be the procedure for developing the descriptors for tree species?
First of all, the curator, the persons who is involved in the breeding or the curation or the curator, he will take all the information, and after that he will call the crop, the experts if available in this group. Then they will sit together and they will standardize the descriptors, the crop experts, the germplasm curator, you can see the person who is having the tree or the germplasm, and the other expert in the field that will sit and will discuss about this descriptor, devising descriptors, even consulting the references of the literature, and along with the crop curator or the expert, they will divide the descriptors.
Okay, any more questions? So there are no more questions, I think. So what I want to convey with such lecture is that NBPGR is exclusively working on various plant species, and NBPGR has devised so many descriptors in different crops. So you can visit the website and get the descriptors from NBPGR portal also. And hopefully they will help if you are working and interested in a particular crop. So with this, sir, I would like to thank you on behalf of all the organizing team and our participants. It was really nice lecture, and it was nice to hear you. And thank you so much for being with us. Thank you so much, sir.
Thank you to all of you. Thank you, Dr. Jyoti, Dr. Mamta, I can see here.
Thank you, sir. Thank you, sir, for the nice lecture. And
Sir, you can unshare your screen now. Jyoti ma'am? Ma'am?
हेलो
हाँ ज्योति मैम
बताइए
तो हाँ तो फिर नेक्स्ट सेशन तो अब
2:30 से हाँ हाँ
ओके ओके
संगीता
ठीक है then we will meet again at 2:30. There is one practical session on in vitro conservation and cryopreservation. So hopefully that will also be beneficial because that is the practical part for the lecture that we had in the morning from Dr. Sandhya Gupta. So I request all of you to be on time and join the meeting. Till now we will break for lunch. Thank you. In the tissue culture and the cryopreservation unit at ICAR and PGR. Her area of specialization is agriculture biotechnology, and she is doing excellent work in tissue culture area with the inclusion of biotechnology. So her nature of work or salient achievements include her research work in tissue culture, food science, agricultural, plant science, and molecular biology. And skills and expertise include probiotics, cloning, food safety, food microbiology, microbiology and safety, microbial molecular biology, food biotechnology, biochemistry, microbial molecular fingerprinting, and tissue culture techniques. And she has held many posts in the past. She has been the assistant professor in Sardar Vallabhbhai Patel University of Agriculture and Technology previously. She has served as a senior scientist at ICAR CIFET Ludhiana. She also has served as a PME in charge of at CIFET Ludhiana. Then one of the big achievement of Madam is that she is the owner of one of the very important patents, which is microbial methods for production of the protein isolates and concentrate from oilseed cakes. And she also has a very good number of publication in high impact journals. So other speaker of today's session is Dr. Ira Vaidya Malhotra. She is a scientist at tissue culture and cryopreservation unit with Dr. Sangeeta ma'am, and her expertise include germplasm conservation of spices, industrial and plantation crops, cryopreservation using different cryogenic approaches, medium-term conservation using in vitro conservation strategies, genetic stability assessment, in vitro conserved crops, then functional genomics, marker development, and genetic diversity analysis in many of the crops, which are well known as well as less known also. And she has very good publication in renowned journals like Plant Cell Reports, Pest Management Science, Industrial Crops and Products, Biotech, etc. Many book chapters. And she is the owner of many awards like SERB Early Career Research Award, then foreign visit grant to attend proteomics course at Functional Genomics Center in Zurich. Then she is the owner of gold medal citation certificate of honor by Himotkar Sahitya Sanskriti Jan Kalyan Parishad. So with this brief introduction, I would like to welcome both of our speakers. So, ma'am, I would request both of you to please start with your lecture, and I hope it will be quite informative for the participants. So please, ma'am.
Thank you, Dr. Mamta, for such nice introduction. I'm sharing my screen.
Yes, ma'am, sure.
Is the screen visible to you all?
Yes, ma'am, it's visible. And
Okay.
it's in presentation mode also.
Okay.
You can start, ma'am.
Okay. So today we are going to talk about in vitro conservation and cryopreservation. Basically, I feel you must be having lectures and practicals on seed storage behavior and seed types. So briefly, I would like to tell for in vitro conservation and cryopreservation, basically we go for the seeds that are either non-orthodox or for the crops that are propagated through vegetative methods. Those crops are basically the priority for in vitro conservation and cryopreservation. And I feel you must be having the background about the types of seeds on the basis of their seed storage behavior. They are basically categorized into two major categories. First is orthodox seeds that can be desiccated to lower moisture contents, and they are not so sensitive to desiccation and freezing as well. So they can be conserved through traditional methods of conservation. Whereas the other category, that is non-orthodox seeds, that include intermediate and recalcitrant seeds, they are sensitive to desiccation and freezing. So this type of seeds can only be conserved through cryopreservation. We cannot conserve them through traditional methods. In addition to this, there are certain crops that either do not produce viable seeds or the seeds are highly dormant. In those cases, if there are methods to propagate them through vegetative modes, then we go for in vitro conservation of those crops. So for these types of crops, as I have told, there are two modes of conservation. One is in vitro conservation. Basically, in vitro conservation is culture and maintenance of plant cells, tissues, or organs as in vitro cultures in form of active cultures, in vitro cultures under controlled atmospheric conditions. And for cryopreservation, it is basically done at ultra low temperatures of liquid nitrogen. The temperature range is between -150 degrees centigrade to -196 degrees centigrade in completely frozen state. So basically, in vitro conservation will be covered by Dr. Ira Vaidya Malhotra, and I will be taking up the cryopreservation. Over to Dr. Ira. good afternoon. So we will be talking about in vitro conservation as medium-term storage strategy for vegetatively propagated crops. So for any in vitro conservation strategy, the first step has to be the initiation of clean aseptic cultures. And for that purpose, we have just given a brief outline of how we go about it. So the first step is the acquisition of the germplasm that is to be conserved. So whatever germplasm our target crop is, for the acquisition, we need to acquire it from an authentic source, and its correct taxonomic as well as passport information should be available. All this information is ensured before we start our work so that we do not conserve something which is misidentified or which is not known, or its in detailed information is not available. For conservation or for in vitro culture initiation, we can use different explants like different plant parts, shoots, nodes, leaves, sometimes roots also, parts of vegetative propagules, floral buds, inflorescence. All of these can be used for as explants for culture initiation. Once we initiate the cultures, before we initiate the cultures, we require phytosanitary certificate for exotic samples, that is samples that have been imported from outside the country. And for this, I'm sure you would have lectures from quarantine. They will explain how, if a phytosanitary certificate is not there, then how those plants are screened in-house, in growth chambers or in screen facilities to ensure their disease-free status. And before you initiate the cultures in vitro, they have to be, their disease status should be known. They should be disease-free, especially from bacteria and viruses. Now, when we go for culture establishment, the explants that we used are first surface sterilized using different fungicides and bactericides. And we clean them, we treat them with these chemicals. Different treatment durations are ensured, and then we culture them onto the pre-prepared sterilized culture medium, usually MS basal salt based medium. Once culture growth is observed and cultures are established, we check them for any bacterial contamination to ensure there is no endophytic bacterial contamination. Now, this gives a screenshot of how you go for culture establishment. This is an example of sweet potato, where nodal plants are used for culture establishment. They are excised, they are disinfected, surface sterilized in the lab, in the laminar air flow, and then they are inoculated onto the culture medium. The second image that you see is of the established cultures. Then after 4 to 12 weeks, depending on the crop that you are working on. In sweet potato, within 4 to 12 weeks, you get good growth. Now, these cultures are then checked for any contamination. If you can streak them onto the bacterial medium, or visually we can inspect them. If contamination is found, those cultures are either rescued. If the material is very less or it is very important, we can rescue the cultures. Or if we have sufficient material, the infected cultures are discarded. And then bacteria-free clones are only maintained in the culture facility. So coming to the conservation strategies, that is medium-term conservation based on slow growth. So when we are talking of in vitro conservation, here what we do is the idea is to increase the subculture duration so that cultures can be conserved for a longer period of time. Now, at this place, tissue culture takes two different approaches. One is tissue culture is used for rapid multiplication, but in terms of conservation, we have to slow down the growth rate of the cultures without affecting their viability so that we can enhance or increase the duration for which they need to be subcultured. There can be different strategies that can be used. One is the physical changes that you can do. For example, we can store the cultures at low temperatures, ranging from 4 to 15 degrees Celsius, and this strategy is usually applied for temperate crops. We can reduce the light intensity. It has been observed that incubating cultures in low temperature along with dark condition has also been found to increase the subculture duration. The culture vessel closures, that is the closure types that you use for your culture vessels, influence the rate of evaporation and gaseous exchange. So we try different culture vessel closures, and the best ones then are then used. The type of culture vessel can be changed. For example, broad-based culture vessels can be used, rectangular ones like magenta boxes, or culture vessels with round bottoms. These again influence the rate of intensity of light that is exposing, the cultures are exposed to, and the rate of gaseous exchange. Another strategy that can be used is the mineral oil overlay, where we can overlay the cultures using mineral oil. And this again reduces the rate of gaseous exchange, further reducing the growth of the cultures. The second strategy that can be done is using chemical changes. That is, we can change the composition of the nutrient medium. Organizers, can you mute them? Yeah, okay. So if you change the culture medium concentration, based on that also the growth rate can be reduced. So we can use minimal medium, where we decrease the concentration of the MS salt, or we can increase the sucrose concentration. In addition, some osmotic regulators can be added to the medium, like mannitol or sorbitol. Then we can use some growth retardants like malic hydrazide. All of these aid to reduce the culture growth. Another strategy that has been employed for slow growth is the in vitro induction of storage organs. This is usually done in crops that naturally form storage organs in field as well. So in vitro induction of bulblets or corms or rhizomes also is helpful in increasing the culture life up to 1 to 3 years under in vitro conditions. Now, one thing that care we have to take during this strategy is that we have to ensure that we are not inducing any mutations in the plants while we are subjecting them to these conditions. So a routine check of their genetic integrity and their physical state, physiological state has to be done to ensure that the treatment given is not hampering their growth, or it is not inducing any kind of changes in those original cultures. So this is an example of the type and size of culture vessels that can be used. For example, glass bottles or jam bottles, as we can call them, are used. These rectangular boxes or magenta boxes are used. Sometimes for conservation, heat sealable bags are used, where we inoculate the cultures in medium in these heat sealable bags, and then we seal them. So cultures can be conserved in them. Some plastic containers can be used, and polypropylene caps are usually employed in our laboratory in place of cotton plugs. And when you see in this image also, the culture with the polypropylene cap is viable, is growing, whereas the culture with the cotton plug has dried up. Now, changing the physical parameters, this is an example of alien species that have been maintained at 8 degrees Celsius, that is low temperature, and the subculture duration has been increased up to 2 years. So again, the temperature has to be lowered in such a way so that no freezing injury is induced in the cells, and the macromolecules of the cells are not damaged. So when we are devising this strategy, care has to be taken for avoiding the damages associated. Now, this is an example in our division that has been done of slow growth by altering the culture medium. For example, overlaying the cultures of Bacopa moneri using mineral oil, where they could induce the increase the subculture duration to 24 months. That is, for 2 years, these cultures could be maintained in vitro without any subculture. This work was done by Dr. Sharma and her group. Another example is of Campharia, where the culture duration has been induced using mannitol in the medium. And the third image that you see is, these are 8 to 10 month old cultures of cardamom and of jojoba. They are maintained on minimal media, that is, the medium concentration has been reduced and minimal hormone or very low hormone concentrations are added, so that the cultures are healthy but they are growing slowly. This is the example of induction of storage organs that has been done in our division in curcuma, where in vitro rhizomes were induced on high sucrose medium, and these in vitro induced rhizomes were then conserved. Similarly, in ginger, done by the group of Tyagi and co-workers in 2006, this has been published. And the third work is by Dr. Hussain and Dr. Tyagi, again in taro, where they have induced in vitro the corms of taro, that is colocasia, and then they could conserve these cultures for long period. Another strategy that has been used for conservation is encapsulation. That is also called the synthetic seeds or syn seeds. Here you encapsulate the growing points of the plant, that is the shoot tips or nodes or whatever is your culture initiation explant. You can encapsulate them in sodium alginate beads, and then you can conserve these beads at low temperature for different time durations. And when you have to grow them again into plants, those conserved beads can be recultured on the regrowth medium, and they have been observed to show good growth and give viable plants. This is an example in ginger where syn seeds have been used for conservation, and in Bacopa, which has been published in the year 2020. So for in vitro conservation, what happens is when you conserve your plants, you have to observe till how long you can subculture your plants. You have to observe that no necrosis or dying is being observed, and rapidly when you observe that 50% of the plants have died out or showing necrosis, then you need to subculture them or rejuvenate them again. So based on these strategies, this is a very good option for medium-term conservation of vegetatively propagated crops. So the next method is for cryopreservation, that is long-term conservation of crops that Dr. Sangeeta will take. So basically, as I have told earlier, cryopreservation is a process in which we conserve the material in completely frozen state, and it is considered that the material is in complete biologically inactive state, and there is no metabolism or cell division is taking place, and hence the germplasm can be maintained theoretically for infinite duration under cryopreserved conditions. So basically, when we talk about cryopreservation, we think just putting the samples in liquid nitrogen is cryopreservation. This is not so. There are some other mechanisms that can also be used for cryopreservation of samples, but in case of plant tissues, the most preferable method is cryopreservation through liquid nitrogen. In addition to liquid nitrogen, we can also use low temperature deep freezers like -80°C in case of animal cells. This is being used for cryopreserving the material. And in addition to this, there is another option, that is solid carbon dioxide, on which we can conserve our sample, and the temperature range is around -79°C. So basically, as I've told, we are going to cover only plant tissue cryopreservation. So this is basically done in using liquid nitrogen, either in vapor phase condition or in liquid condition. So this also I have discussed. The priorities for cryopreservation are those species that produce non-orthodox seeds or that are propagated through vegetative modes. And these are the examples of explants that can be used for cryopreservation. Like, we can conserve the seeds itself. We can also, if seed is not responding, then we can go for excising the embryos, and these embryos can be cryopreserved. In addition to the embryo, we can also try embryonic excess. In case of temperate crops that have tendency to produce dormant buds, these dormant buds can also be a target of cryopreservation. In case of vegetatively propagated crops and for long-term conservation of in vitro cultures, we can go for conservation of shoot apices or apical meristem of a plant. And in addition to this, where there are some demand or there is need or there is asynchronous flowering, then we can also go for pollen conservation. So cryopreservation involves a number of steps. In this slide, we have listed those steps. First of all, we need to have an explant or sterile tissue. The second step involves the pre-treatment and cryoprotection steps. The third step is freezing. Fourth step is basically storage of the treated samples. Fifth step is when we want to regenerate the samples, we have to go for thawing. Then after thawing, we have to regenerate the samples using the optimized mechanisms. And these are the basic steps that are being followed for cryopreserving any sample. And in addition to this, we also need to check the survival and viability of the conserved sample time to time. So as I have listed these steps, the first step is the explant or starting material. So we have to be cautious while choosing the starting material or explant, and it has been shown through studies that the explant should be in actively growing state, and it should be and actively dividing the material is preferred over dormant tissues because actively dividing material contains smaller size cells and there are less vacuoles and therefore less water content. So it is preferable that we pick the tissue from actively dividing the plant or cultures. And it has been basically also observed that the size of explant which we are going to conserve is also important. If we take larger explants, then it is difficult to homogeneously desiccate the entire sample, and due to that there may be certain zones of ice crystal formation, and that finally affect the viability. So larger explants are usually not preferred. But at the same time, too small explants are also difficult to isolate, and sometimes mechanical injury may take place, and these smaller explants sometimes may not survive and regenerate into complete plantlet. So we have to make a certain equilibrium between large and small size, and basically it has been shown through studies the appropriate size of explant is around 0.5 to 1 mm. Once we have the explant, then the next step is pre-treatment or cryoprotection. For pre-treatment, generally the samples are pre-treated using different methods. Pre-treatment is basically, say for example, for temperate crops that can tolerate low temperature. We go for preconditioning the cultures. We keep the cultures at low temperature for certain durations, like we can keep the cultures at 10 degrees centigrade for up to a month. So it prepares the sample for further freezing. When we are going to cryopreserve the sample, it will prepare the sample for that harsh treatment. And in case of tropical crops, generally in case of tropical crops, generally high sucrose concentrations are used in the culture medium so that some osmoticum is created and water content of the tissues is reduced. In addition to this, there are certain procedures we have to use. The cryoprotectants are the ground that can prevent the damage that is caused during freezing due to ice crystal formation. And what these cryoprotectants do, they basically lower the freezing temperature or super cooling point of a solution, and it induces the formation, it increases the density of the solution, and therefore it prevents the formation of ice crystal. In addition, in place of ice crystal formation, amorphous state is formed, and that is not injurious to the cells. itself. The examples of cryoprotectants include DMSO, glycerol, ethylene glycol, etc. Basically, the cryoprotectants are of two types. First one is permeating or penetrating cryoprotectant, and second class is non-permeating or non-penetrating cryoprotectant. Permeating cryoprotectants are basically smaller size molecules and that can cross the cell membranes. And the examples of permeating cryoprotectant include glycerol and DMSO. And what these permeating cryoprotectant do, basically they have the capacity to cross the cell membranes. When there is concentration gradient, they can cross the cell membrane and they provide intracellular protection to the cells. And basically, as I have told, cryoprotectants generally decrease the freezing point of a solvent or solution, and they also prevent cell damage due to high electrolyte concentration. And they also form linkages with the electrolyte molecule that are present in the cell cytoplasm, and they substitute the water that is present inside the cell. So in addition to this, these penetrating cryoprotectant has also the capacity to stabilize the lipid membranes by formation of hydrogen bonds with the lipids that are present in the membrane. And this is very important when the cell is dehydrated to severe levels. In addition to these penetrating cryoprotectant, there is another category of cryoprotectant, that is non-permeating cryoprotectant. Basically, these cryoprotectant cannot cross the cell membrane. And how do they provide the cryoprotection? They provide the cryoprotection from the outside of the cell, and they create an osmotic gradient in the cryopreservation solution that is present out of the tissue. And in this way, they provide the cryoprotection. And due to this osmotic gradient, there is loss of water from the cells, and the water flows from inside of the cell to outside, and this leads to dehydration of cells, even in the presence of permeating cryoprotectants. So these are some examples of permeating and non-permeating cryoprotectant. You can see the permeating cryoprotectant generally involve organic solvents like ethylene glycol, DMSO, polypropylene glycol, acetamide, formamide. And non-permeating cryoprotectants are majorly sugar molecules like glucose, sucrose, trehalose, raffinose, PVP, mannitol, etc. And basically, for cryoprotection purpose, it is recommended that a single cryoprotectant is not used, and a mixture of permeating and non-permeating cryoprotectants are recommended. After once we have extracted the once we have prepared the explant and we have done the pre-treatment and cryoprotection, the next step is freezing. Freezing is very important, and there are different modes in which freezing can be done. Freezing can be either through slow cooling mode, or it can be done with in form of rapid or ultra rapid cooling. What is slow cooling? In slow cooling, basically the temperature is reduced in stepwise manner. It is not instantaneous. The temperature is gradually reduced, and for most of the methods of slow cooling, which is followed most commonly, is to reduce the temperature at a rate of around 0.5 to 2 degrees centigrade per minute, up till a pre-freezing temperature of around -40 degrees centigrade is achieved. Once the pre-freezing temperature of around this -40 degrees centigrade is achieved, then the sample is rapidly immersed in the liquid nitrogen, where the temperature is -196. So we also call this method of slow cooling as two-step method or stepwise freezing. And this can be done with the help of a sophisticated instrument that is known as programmable freezer. And this programmable, we can set up a program in this programmable freezer that can reduce the temperature of the samples at a controlled rate as per our wish and as per our optimized condition. but this is a quite costly equipment and it requires the liquid nitrogen. So there are certain economical options to this programmable freezer. In programmable, the advantage of using programmable freezer is that we can set up any cooling rate, whatever we wish. If we want to cool if we want to cool the sample at a rate of 0.5 degrees centigrade per minute for initial 10 minutes, then we want to switch at a rate of -1 degree centigrade per minute, then it is possible to set up that type of program in programmable freezer. In these type of boxes, it is not possible. The rate of cooling is fixed. Usually the rate of cooling is around 1 degree centigrade per minute. We cannot set up a stepwise program, or we cannot set up the cooling temperatures at our wish with the help of these cooling boxes. And these are the examples, two examples. These are proprietary items. One is Mr. Frosty, that is available from the firm Thermoscientific. And in this, we can use 1 to 5 ml cryo tubes. And as I have mentioned, the rate of cooling in this box is around 1 degree centigrade per minute. It will cool the samples at a rate of 1 degree centigrade per minute. And the capacity of this particular box is around 12 to 18 tubes per box. And between the isopropyl alcohol is filled between the two layers of this box that provide the cooling to the inner samples that are present in the cryovial. Similar to this, Mr. Frosty, another proprietary item, that is Corning Cool Cell, is also available in the market. And this also has a cooling rate of 1 degree centigrade per minute when we put our samples in the cryovial in this box, and this box is kept in -80 degree freezer. So we can, if we do not have programmable freezer, we can go for these type of boxes that are comparatively economical. Once the freezing is achieved, then the next step is storage. Storage is also very important step in cryopreservation. And as I have told, basically the most preferred method of storage is the storing of samples in liquid nitrogen. And for storing purpose, we have two options. We can either store the samples in vapor phase of liquid nitrogen, or we can also store the samples in liquid phase of liquid nitrogen. These are the example of two different tanks. First one is vapor phase storage tank, and second one is liquid phase storage tank. In vapor phase storage tank, the cryo boxes containing cryo vials are placed above a platform. You can see a platform. Here is a platform that is placed on a riser, and liquid nitrogen is filled below this platform. And the sample does not come in direct contact of liquid nitrogen, and the samples are stored in vapor phase, in vapors of liquid nitrogen above this platform. And there is a temperature gradient throughout the tank. At lower levels, there will be lower temperature, and as we rise towards the lid, the temperature increases. And the temperature range that is present within this vapor phase tank is -150 to -180 degrees centigrade. The second option that can be opted is liquid phase storage. In liquid phase storage, you can see here, these are the cryo boxes, and the entire tank is filled with liquid nitrogen, and the boxes are directly immersed in liquid nitrogen. And in this condition, the temperature throughout the tank is around -196 degrees centigrade. For basically the tissues which are having high moisture content, we prefer liquid phase storage. Like for meristem, shoot apices, and dormant buds, we go for liquid phase storage. And the advantage of liquid phase storage is that the temperature throughout the tank is uniform. But there are certain problems associated with liquid phase storage that are, first one is it is difficult to handle the samples under liquid phase storage. And sometimes it may happen that this LN, liquid nitrogen, may enter into the storage tubes or cryo vials. And if this liquid nitrogen enters into the storage tubes or cryo vials, there are risks of sample contamination and cross-contamination between the samples due to microbes. And sometimes when when high temperatures are there, this LN may evaporate and expand, and sometimes the cryovial may explode on increasing the temperature. And in liquid phase tanks, the evaporation rate is comparatively higher. That increases the cost of storage. In vapor phase storage, all these problems are solved because the samples are not in touch of liquid. So there is no chance of cross-contamination. And it is also easy to handle the samples under vapor phase storage. Preferred explant for vapor phase storage are seeds that may be orthodox or non-orthodox, embryos, embryonic axis, and pollens. And problem associated with vapor phase storage is that as I have mentioned, that there is a temperature gradient that is present in the tank. Uniform temperature is not there throughout the tank. So you have to monitor the temperatures of the tank very cautiously. So for cryopreservation, separate kind of vessels, vials, and storage materials are required. This slide gives a glimpse of the vials or storage systems that are required for cryopreserving any sample. Like these type of cryo vials that can sustain the ultra low temperature of liquid nitrogens are required. These vials are available in different sizes, like 1 ml, 2 ml, 4.5 ml, or 50 ml, and choice of vial depends upon the size of the sample which we want to conserve. Like for shoot apex or meristematic tissue or embryos or pollen, we can go for smaller vials. 1 or 2 ml vials are sufficient to store large quantity of sample. But for large size seed, we may require larger size vial, that is 50 ml vial. And for placing these vials properly, there are separate kind of systems are available. I am particularly giving the example of the tank or inventory system that is being used at our place. And we have these kind of cryo sticks for smaller vials. These cryo sticks are used. These cryo vials are put on these cryo sticks, and these cryo sticks are then placed in these canister. These are cryo vials. This type of sticks, these are called as cryo sticks, and these square boxes are called canister, basically. So when smaller vials are used, these vials are placed on cryo sticks, and these cryo sticks are then placed in a canister. But when large sized vial like this 50 ml vial is used, then we place these vials directly in the canisters. There is another option, like if we are going to conserve the bud wood or wood sticks, then the vial size should be large enough to hold the entire wood stick. For those, we have these kind of polyolefin tubing that can be customized in size as per our need. In addition to this, this is basically for vapor phase tank that is available at our place. For shoot apex, I have told we are using liquid phase storage tanks. For those liquid phase storage tank, the cryo vials are basically put in these cryo boxes, and these cryo boxes are then placed in these cryo trays, and then these cryo trays are placed in the cryo tank. And this type of cryoprotection equipment is always necessary when we are going to store the sample, or when we are going to retrieve the sample, or when we are dealing with large volumes of liquid nitrogen. It is always necessary that we put on these cryoprotection equipment. When we want to retrieve the sample after certain storage duration, thawing is also important. And generally nowadays rapid thawing is being done. And for rapid thawing, what is done, the sample, the cryo oil is removed from the tank, and it is immediately immersed in the beaker or water bath that has a temperature of a range of around 37 to 45 degrees centigrade for 1 to 2 minutes, so that rapid thawing is achieved. And in addition to this, from time to time, it is always necessary to check the survival percentage or viability of the stored sample. It depends upon the method of survival or viability testing, always depends upon the explant and how what is the optimized procedure for its regeneration. Like if it is shoot meristem or shoot apex, then viability is basically tested through culturing this shoot apex on optimized culture media, and regrowth is observed. But for seeds, we can go for directly germinating the seeds on filter paper. So on the basis of different parameters and different procedures and different steps that are being followed during cryopreservation of any sample, a number of different techniques have been devised. And these are the major techniques which we are going to cover. We can, there is a simplest technique is desiccation freezing. Desiccation freezing is nothing but in this technique, we directly desiccate the samples, and this desiccated sample is directly frozen. In addition to this, I have mentioned that we can go for two-step freezing. It is also a method of desiccation freezing, but in this case, the temperature is gradually decreased. In addition to this desiccation freezing and two-step freezing, certain other techniques are also available, like encapsulation dehydration, vitrification, droplet vitrification, and encapsulation vitrification. In this encapsulation dehydration and vitrification, these techniques are basically categorized in the category of rapid freezing techniques. And this droplet vitrification, basically this is ultra rapid freezing technique. So this is a screenshot of desiccation freezing technique, how this is basically done. We have shown this with the example of oil palm embryo. For desiccation freezing, what is basically done, first of all, we take the seeds and extract the oil palm embryos aseptically. After this extraction procedure, we air desiccate the oil palm embryos in laminar air flow for 3 hours. For desiccation, we can either use, if the sample size is small, then we can go for direct air flow for desiccation under airflow of laminar flow. Or if the seed size is larger, then we have to use silica gel for desiccation. And once the sample is desiccated, it is put into the cryo vials, and then this cryo vial is plunged into the liquid nitrogen. So basically, desiccation freezing involves simply desiccation of simple desiccation of the sample and then freezing. And when we want to extract the samples, thawing is basically done at 40 degrees centigrade for 2 minutes, and then regrowth of this oil palm embryo is basically done on optimized regrowth medium. After desiccation freezing, the another technique that can be used for cryopreservation is encapsulation dehydration. If desiccation, first of all, we check whether our sample is responding for desiccation, simple desiccation freezing or not. If it is not responding to that, then we can go for encapsulation dehydration. Encapsulation basically provide an external coating to avoid direct injuries to the tissue or sample. So we can go for encapsulation dehydration. In this slide, you can see the basic procedure of encapsulation dehydration. In encapsulation dehydration, we have shown in this particular slide with the help of shoe tip. In encapsulation dehydration, the explant is taken and then it is encapsulated in calcium alginate beads. Generally, the concentration of calcium alginate bead is at around 2 to 3%. After encapsulating in calcium alginate bead, primary desiccation is done with the help of osmotic dehydration in presence of high sucrose concentration, and generally the concentration of sucrose in this osmotic dehydration media is around 0.75 molar. And after that, when after initial osmotic dehydration, this osmotic dehydration we have to optimize which duration is optimum for osmotic dehydration. Even the concentration of sucrose can also be varied, like it may a range of 0.75 molar to 1.25 molar sucrose can be tested, and desiccation during duration of 1 to 3 days may also be tested to devise the optimum duration. After osmotic dehydration, the next step is air dehydration of these beads. And air dehydration, generally air dehydration duration has also to be optimized, and it ranges from 4 to 6 hours under standard condition of laminar airflow. And the moisture content, generally final moisture content that is reported to produce viable regrowth is around 22 to 25% moisture content. So we have to desiccate the beads so that we achieve around 22 to 25% final moisture content. Once this moisture content is achieved, these desiccated beads are placed in cryovials, and then this cryovial is placed in liquid nitrogen. So basically in this technique, the beads are first placed in cryovial, and these cryovials are placed in directly plunged in liquid nitrogen. This technique is called as rapid freezing technique. And in this technique, the tissue sample does not come in direct contact of liquid nitrogen. Instead of that, the cryovial comes in the contact of liquid nitrogen. And it is since the cryovial is directly plunged in liquid nitrogen, the rate of cooling are very high, and this is basically a rapid freezing technique. And when we want to extract the samples, again we have to go for thawing for 2 minutes at 40 degrees centigrade, and then we put the samples on regrowth medium to check the viability of the samples. Another variant that is available in rapid freezing technique is vitrification technique. In encapsulation dehydration, we are basically going for dehydrating the protected sample or protected tissue that is present in a protective layer or protective coating. But in vitrification, it also involves the use of cryoprotectants. In simple desiccation freezing and encapsulation dehydration, these type of technique does not involve the use of any cryoprotectant, and they are simple desiccation techniques. But in vitrification, we make use of cryoprotectants to produce vitrifying conditions, and vitrification solutions are basically used for this. And in this technique, what is basically done, we extract the first of all, we extract the explant or shoe tips, whatever be our explant. Then preconditioning or pre-culturing of the explant is done on 0.3 molar sucrose medium. Then we treat these samples with high sucrose containing loading solution. Then after that, the next step is treating the sample cryoprotection or treating the sample with a mixture of different cryoprotectant. The most commonly used cryoprotection solution is plant vitrification solution 2 or PVS2. This is the most commonly used cryoprotecting solution in different plant tissue culture conservation practices. And PVS2, basically it contains glycerol as I have told earlier, that it is recommended that we use a mixture of permeating and non-permeating cryoprotectants. So PVS2 basically contains glycerol, DMSO, and sucrose. It is a mixture of permeating and non-permeating cryoprotectant. So first of all, loading solution is osmotic protection is done with the help of loading solution that contains only sucrose. Then next step is cryoprotection with the help of plant vitrification solution 2. After cryoprotection, the samples are placed in cryovials, and these cryovials are plunged in liquid nitrogen. Again, this is a technique of this technique is also an example of rapid freezing technique. And when we want to extract the sample, again, we have to thaw the samples at 40 degrees centigrade for 2 minutes. Then unloading is done with the help of high sucrose concentration containing solution, and then regrowth is achieved on optimized culture media. In this figure, you can see this is an example of Dioscorea rotundata, and you can see basically this slide shows the growth of control, and this shows the growth of vitrification control. Vitrification control is basically when we take the samples after this step, after vitrification step. As I have told that these cryoprotecting agent are used for providing cryoprotection, and they lead to dehydration and modification of the cell microenvironment. They sometimes they have reported to be toxic to the cells also. So we always check the growth of vitrification control also before freezing. And the third shows the growth of cryopreserved nodal explant, and this plantlet is this is the fully grown plantlet from these cryopreserved nodal explants. The next variation in these cryoprotection technique is encapsulation vitrification. Encapsulation vitrification is basically a mixture of the technique of encapsulation with vitrification or cryoprotection. So as I have mentioned that in the technique of encapsulation dehydration, we just go for encapsulating the sample and then dehydrating or desiccating it directly. But in vitrification, we provide cryoprotection also. So this flow diagram shows the different steps of encapsulation vitrification techniques. Once the explant is ready, then we go for encapsulating the explant in calcium alginate beads. And then these calcium alginate beads are not desiccated through air desiccation or silica desiccation. We desiccate them stepwise as has been done in the vitrification technique. These beads are first of all put in loading solution. That is, osmoprotection is given. After osmoprotection, then these beads are placed in plant vitrification solution 2 that contains a mixture of different cryoprotectants. Then these beads are placed in cryovial, and this cryovial is plunged directly into the liquid nitrogen. And rest of the steps are similar to the encapsulation dehydration or vitrification techniques. Like when we want to extract, we have to thaw the samples again. So these three techniques that includes encapsulation dehydration, vitrification, and encapsulation vitrification. These are categorized as basically rapid freezing techniques, because in these techniques we place the samples in cryovials, and then this cryovial is plunged into the liquid nitrogen. The sample does not come in direct contact of liquid nitrogen. So these are rapid freezing technique. Recent advancement in the cryoprotection techniques involves ultra rapid freezing techniques. And these are the examples of ultra rapid freezing techniques that include droplet vitrification technique, use of different types of cryo plates like V cryoplate technique and D cryoplate technique, and cryo mesh technique. In droplet vitrification, basically, after isolation of shoot tips, the shoot tips are pre-treated in pre-treated on solid culture medium that contains high concentration of sucrose, that is exactly 0.3 molar sucrose. We can vary the duration of this pre-treatment. After pre-treatment, again, and then at the step of cryoprotection, given for optimized duration. After cryoprotection, the sample is placed onto these aluminum foil strip. In place of cryo vial, the sample is placed on these aluminum foil strips. On these aluminum foil strip, first of all, we place a drop of PVS2 solution, and on these PVS2 drops, we place the shoot tips. And then this aluminum foil is directly immersed into the liquid nitrogen. So in this particular technique, you can see that the sample comes in direct contact of liquid nitrogen. The sample is in direct exposure of liquid nitrogen. And after freezing these aluminum strips, these strips are placed in cryo vials. And when we want to extract the samples, again, the rest of the steps are same. Rapid thawing is done at 40 degrees centigrade for 2 minutes. Then washing and unloading is done in high sucrose solution. And then culture of the explant is again done on optimized culture medium. If anyone of you if someone of you has any doubt, you can ask, you can stop me in between also.
Yes, I'm sure.
Are you people following what I am telling?
Yes, ma'am.
Yes,
ma'am.
If you have any query or doubt, you can ask. I have a recorded clip of DV technique. I'm going to share that clip, the video clip with you, so that you have a clear idea how the different steps or different procedures are done in droplet vitrification technique. Just let me know if the clip is visible to you.
Okay, ma'am. Once you start playing, we will confirm.
Is the clip visible to you?
Clip is visible, but still not playing, ma'am.
Actually, I have paused the clip. I just wanted to inquire whether it is visible to you all.
In its icon, we are able to see.
No, madam, not visible at all.
Not, you cannot see.
Only the screen is there.
Okay. Let me check. Now?
No, ma'am.
No, madam.
Again, the screen is visible, not the.
Yeah, but the
Probably it
video is not
about-
playing.
Yes, yes.
It has started to play.
Now you
ma'am.
can see it.
Yes, ma'am.
Yes, madam.
Yes,
Yes,
ma'am.
yes.
So it is visible to you all.
Yes, yes.
Yes,
Yes.
ma'am.
Okay, okay. So basically, this is the setup of laminar airflow for any cryopreservation technique. For vitrification, I have mentioned, we require 3 types of solution. One is loading solution, another one is PVS2 solution, and unloading solution. So we have to place all the required solution in laminar airflow. In addition to this, the explant. In this particular clip, the explant is shoot tip. Okay. These are the in addition to this shoot tip, we also require cryo vials that are autoclaved before use. We also require filter papers. This is cryo box basically. For droplet vitrification technique, this is the basic setup. In the cryo box, in this thermocol box, we put this cryo box, and in this cryo box, we place the cryo vials with opened lid. Okay. So we have now poured the loading solution into these small Petri plates. Then the shoe tips are carefully taken from the Petri plate and these are placed into the loading solution. This clip is visible to you all. I am playing the second clip.
No, ma'am.
Again, it is not. Can you see the clip now?
No, ma'am.
Is it visible now?
No,
Not
ma'am.
yet?
No, ma'am, no.
No, ma'am.
Can you see the clip now?
No, ma'am. It's not playing.
Video is visible, but it is not playing, or
No, ma'am. Video is also not visible. Only a one black screen is coming, gray screen.
I don't know how to share. Now?
Yeah. Now it's playing, ma'am.
Okay. So once we have placed the sample in loading solution for the required duration, after that, this loading solution is removed with the help of a dropper. And care has to be taken so that we do not suck up these shoot apices, or we do not injure these shoot apices during removal of this loading solution. After loading solution is removed, we put the cryoprotection solution, that is PVS2, into the petri plate containing the explants, and it is incubated for the required duration. After required incubation is completed, we again remove this PVS2 solution from these petri plates. You can see this aluminum foil strip here, this one.
Yes, ma'am.
So once the required incubation is completed in PVS2 solution, we place 1 drop of this PVS2 solution onto this aluminum foil. This video is basic demonstration of droplet vitrification technique. So we put 1 drop of PVS2 solution onto this aluminum foil. Then we carefully place the shoe tips on to this aluminum foil that contains the drop of PVS2. PVS2 is basically a mixture of different cryoprotectants. Generally, 10 shoe tips are placed on a single aluminum foil strip. After placing the shoe tips on this aluminum foil strip, this aluminum foil strip is directly placed in the liquid nitrogen. So you all can see that the samples are coming in direct contact of liquid nitrogen. So in droplet vitrification technique, the samples are basically placed on aluminum foil strip, and this foil strip containing the samples is directly plunged into the liquid nitrogen. So this technique is basically considered as ultra rapid freezing technique, because the sample is coming in direct contact of liquid nitrogen, it is immediately frozen. Okay. I hope the basic difference between rapid freezing and ultra rapid freezing technique is you all can understand what is the basic difference between rapid and ultra rapid freezing techniques. We can have an interaction so that I can understand whether you people are following or not.
Friends, please answer. Are you able to follow what ma'am is saying? Please try to interact also.
Or I assume that everything is clear to you all.
In the chat box, ma'am, they are replying, yes, they are following.
Okay, they are following, then it's fine. So basically in vitrification and encapsulation vitrification techniques, the samples are placed in cryovial first, then cryovials are closed, and this cryovial is placed in liquid nitrogen. In those techniques, the vial comes in contact of liquid nitrogen and the sample is gradually frozen. First of all, the solution that is present in the cryovial is frozen, and then the sample is frozen. So those techniques are basically called as rapid freezing technique, and in ultra rapid freezing technique, the sample comes in direct contact of liquid nitrogen. So there are very high rate of cooling in ultra rapid freezing technique, and they are considered more efficient in terms of viability retention. Okay, so I think droplet vitrification technique is clear to you all. In droplet vitrification technique, these aluminum foil strips are used, and explant or shoot tips are placed on PVS2 solution on these aluminum foil strips, and then these foil strips are directly plunged into the liquid nitrogen. So you have seen that when we are handling, it is very tricky to handle these aluminum foil strips that contains the samples in liquid droplet. So some kind of expertise is required to handle such technique. So to ease out the difficulty of this particular technique, another variation has been devised, that is V crab plate technique and D crab plate technique. So in V and D crab plate technique, basically special type of crab plates are used that contains these wells, and on these wells the shoot tips are affixed with the help of generally calcium alginate. So in this slide shows the basic procedure of D crab plate technique. In D crab plate technique, once the shoot tips are excised, after that these shoot tips are placed onto this special kind of cryo plates that contains 10 wells of exactly similar sizes, and these shoot tips are placed in these wells, and they are adhered to these wells with the help of calcium alginate. So for adhering these shoot tips to cryo plate, first of all shoot tips are placed, then sodium shoe tips, the sodium alginate solution is poured, and onto this sodium alginate solution, we overlay these cryoplate with calcium chloride, 100 millimolar calcium chloride solution, so that calcium alginate formation takes place. And with the help of calcium alginate polymerization, these shoe tips are fixed to these cryoplates. So it is comparatively easy to handle the fixed shoe tips in comparison to floating shoe tips. So this is comparatively easy and efficient technique as we compare it to droplet vitrification. Once the shoe tips are affixed onto these cryoplates, then again the similar procedure is followed. We go for osmo protection in loading solution. Then we go for, I think this is V cryoplate. Yes. This is V cryoplate. After osmo protection, again cryoprotection is being done with the help of PVS2 solution. In this case, we do not have to individually pick up the shoe tips. The first step is affixing these shoe tips onto the cryoplates. Then this entire cryoplate is transferred from one solution to another solution, and it is comparatively easy to follow the steps in this particular technique. So after osmo protection, cryoprotection is done. After cryoprotection, again these cryoplates are directly plunged into the liquid nitrogen, as we have done in droplet vitrification technique. And when we want to extract the sample, again rapid thawing is done, and then regrowth is basically seen whether the samples are viable or not. Another variation of these cryoplate technique is decryoplate technique. When we use the word V indicates vitrification, and vitrification means the use of cryoprotection. Okay, and when we generally use the word D indicates dehydration. So in decryoplate technique, the shoe tips are adhered to these cryoplates, and then osmo protection is basically done in a loading solution that contains high sucrose concentrations, and after that these are desiccated in laminar airflow to achieve the optimum moisture content of the shoe tips, and then without any cryoprotection, these dehydrated shoe tips are directly plunged into the liquid nitrogen. So basically droplet vitrification and D and V cryoplate techniques are ultra rapid freezing techniques. Another variation, basically the problem with V and D cryoplate technique is that both of these techniques require these specialized kind of cryoplates that are not easily available, and there are no commercial firms or companies that are providing these type of cryoplates. So it is difficult to obtain these cryoplates. So another variation of these cryoplate has been developed that uses steel mesh in place of these cryoplates. This is the technique is entirely similar to V cryoplate technique, but in place of cryoplate, this technique involves the use of a stainless steel mesh, and this can be cut down from a stainless steel sheet, and it can be optimized as per our requirement, as per the size of the cryo vial. We can cut down the strips. Okay, these are the major basic techniques of cryopreservation. If I want to summarize, we can summarize. Basically, they can be classified into 3 categories. Simplest one are simple desiccation and then freezing. The second category is vitrification, that is cryoprotection and freezing. Third one is cryoprotection and direct immersion of samples in liquid nitrogen. Basically, these 3 variants are available, and we can try and test these techniques as per our requirement and as per the explant which we want to conserve. I want to talk about pollen conservation and pollen cryopreservation also. This is a separate kind of explant or material. And certain considerations should be kept in mind when we want to conserve the pollen. Like we should collect the pollens from deceased anthers, and proper time of collection of anthers is important, and generally the anthers are collected in the morning on bright sunny day at the time between 8 to 10 a.m. And when after collection of the anthers, we have to check if proper decency is there or not. If incomplete decency is there, then we have to put the anthers in a table lamp that contains a bulb, so that the anthers are decised, and we can extract the pollen. And these pollen are collected with the help of needle onto clean butter papers. And in certain species, there are sticky type of pollens, and for those sticky pollens, we can use organic solvents. So basically, this flow diagram shows the basic procedure of pollen collection and conservation. Like if anyone of you want to get your sample conserved at our place, then you should collect the pollen properly. And if you want to transport them to our place, then they should be transported in desiccated state. So basically, it is recommended that after collection of the pollen, you desiccate them to optimize duration, then place or pack them in aluminum foil, and these foil are then placed in cryovials, and then you can ship these pollens to us so that we can go for cryobanking of these samples. But be sure that pollen viability has been checked after desiccation, and desiccation has been done for optimized duration so as to maintain the best viability of the pollen. For viability testing, there are different methods. We can go for, I think TC test has been described in the practicals on seed germination methods. So for viability testing, either we can go for direct in vitro germination of pollen samples. Different types of media compositions are available. We can check the germination of pollen on these pollen germination medium. And we can also go for TTC staining or FCR testing that produce certain specific kind of colored compound or fluorescence that indicates the viability of pollen. And in the figure given at the bottom of this slide, you can see these are the TTC stained pollen. The viable pollens are stained in pink, and the pollens which are not stained, that indicates these pollen are dead. This particular slide, this particular figure shows the germination of pollen. So these are, to sum up, these are the basic steps that are being followed for cryobanking or cryopreserving any solution, any sample at our place. In addition to, though I have given major examples of shoot apices, in addition to shoot apex, I have also told that we can also conserve the seeds, embryos, dormant buds, etc. So if that kind of samples are there, then first of all, these are the basic steps that are being followed for cryobanking of a sample. We have to excise this explant from the sample material, and it is recommended if you want to transport the sample from a distant place for cryobanking, you should not carry the excised material, because during transportation it may lose moisture, and in case of, particularly in case of non-orthodox seeds, if the samples are desiccated during transportation itself, it affects the viability of the seed. So it is always recommended that you collect the samples, fruit samples, and transport the fruit itself that contains the seed. And after the material is received at our place, then we extract this explant, and after extraction we estimate the fresh moisture content of the sample. Then if certain kind of dormancy is there, then we have to optimize the germination procedure of that seed, and viability and germination ability of the sample is tested. Then after that we have to standardize the desiccation protocol. And once we have optimized the desiccation protocol, then we have to optimize the freezing protocol. And after optimization of desiccation and freezing protocol, we place the samples in the cryo tank. And during placement of these sample in the cryo tank, proper maintenance is required, and a proper record and database needs to be maintained. So these different kinds of cryotanks are available in the market for cryopreservation of the samples. At our place, we have this LN station that can store around 6,000 liters of liquid nitrogen. And because the samples are maintained in liquid nitrogen, so you should have a provision of continuous liquid nitrogen availability as and when required to refill these storage tanks. So for the purpose, we have this LN station or LN storage tank that is of 6,000 liter capacity. In addition to this, this is the photograph of vapor phase liquid nitrogen storage tank. And in addition to this, for the transportation purpose, we also need to have dry shippers. These dry shippers basically contain some absorbing material, and that has capacity to absorb the liquid nitrogen. And then we can place the sample in the slots available within these dry shippers. The advantage of using dry shipper is that liquid is entirely absorbed in the absorbent material, so there is no chance of spillover or some injury due to liquid nitrogen. As I mentioned earlier, that two types of tanks are available: liquid phase tanks and vapor phase tanks. This diagram shows the internal structure of a liquid phase tank. You can see this tank contains these trays. On these trays, cryo boxes can be placed that contains the cryo vials. In addition to this, there can be another type of tank that is vapor phase tank. In vapor phase tank, at our place, we are having these vapor phase tank. You can have a look or glimpse of these tank. Like when we open the tank, the liquid nitrogen used to get evaporated, and it is difficult to visualize the samples under these liquid nitrogen vapors. And this is the internal structure of this vapor phase liquid nitrogen tank. In this vapor phase liquid nitrogen tank, these canisters are placed above a platform. These are the canisters, and these are placed above a platform. And in these canisters, we put the cryosticks that contain the cryo vials. And in this particular tank, we need to specify the location of each and every sample very specifically and very precisely. Like you can have a idea how this is done. Like I have told, we have these kind of tanks, and each tank is having a rotating platform that has 6 different quarters. You can see these quarters. I have shown these quarters as 1, 2, 3, 4, 5, 6. This is separated into 6 different quarters. This photograph shows 1 quarter. Can you see this? This is 1 section or 1 quarter of this rotating platform out of those 6. And on each of these quarters, we can place the canisters in separate rows. You can see in first row, there is provision of 1 canister only. In second row, we can place 2 canister. In third row, we can place 1, 2, 3, 4 canisters. and so on. Okay. So for specifying the location, you need to specifically give some code name to these tanks, like we are having 6 tanks at our cryogene bank. So we have named these tanks from A to E, A, B, C, D, E, F. So if we want to specify the location of a particular sample, then we need to know that the sample is placed in which tank. Like if you want to place the sample in tank 1, then we'll put first of all A to that sample location. And in this A tank, if we are placing the sample in its first chamber, then we will place A1. A1 indicates that the sample is placed in the first tank on its first chamber. After providing this information, we used to put the information about the canister and about the row and the canister. As I have mentioned that on each of the chamber, on each of these chamber, there are separate rows. Like this is first row, you can see this is first row, this is second row, and so on. So we have to next place the number of row. For the purpose, we have used the smaller alphabets like A, B, C, D, E, and F. These indicates the row number. So A1 means, capital A means first tank, first chamber, smaller a means first row, and this numeric one means first canister. Okay. If on my copy I have written the sample is placed on A1, that means the sample is placed in the first tank on its first chamber, in the first row of the first chamber, and in first canister. Okay, so this needs to be noted very precisely. Sample location should be very precisely noted, otherwise there are chances that if proper record or proper maintenance is not done, we may lose or we may dislocate the sample. So basically, as I have told that proper location needs to be mentioned on each and every sample vial, and a record of this needs to be maintained in form of either register or in soft copy, and it is recommended that we keep the record in both the forms. If one form is lost, then we have a backup in another form. So it is recommended that the record should be maintained in hard copies as well as soft copies, and proper location and proper information needs to be recorded and maintained for each and every sample. And in our database, it is always necessary that we should clearly mention the name of the species. Specific accession number is assigned to each and every sample, and this accession number should be correctly recorded and recorded in the register and on the sample vial itself. And in addition to this, it is absolute necessary that we record the exact location of the sample in our notebooks and on sample vial also. Okay, with this I would like to end this particular practical. If you are having any question or query, you can ask.
Thank you so much, ma'am. A few questions I can see in the chat box. I hope you also can see, ma'am.
I'm checking, I'm seeing.
Okay, so the first question is that Dr. Devarajan from TNAU, he has asked that about the loading and unloading solution details.
Loading and unloading solution?
Details.
Details. Composition he is asking?
I think that is what he means.
Yes, ma'am. It's about the composition of the loading and unloading.
Okay,
Yes, yes.
basically loading solution specific composition. If you provide me your email details, I can share with you, or you can check. There is one training manual available on our website. 8th international training on this in vitro conservation and cryopreservation, in which the composition of each and every solution is given there. Basically, the loading and unloading both the solution contains high concentration of sucrose. Unloading generally 1.2 molar sucrose and loading 0.75 molar. High sucrose concentration containing loading solutions are these. Specific concentration you can check in that training manual.
Okay, ma'am. Thank you. Ma'am, one more question.
Yes.
While writing the name of the species and other details in the cryo vials or while keeping in the liquid nitrogen container, storage vessels,
Yes,
what type of material you are using to write the label?
we have cryo markers from the firm Sharpie or Stadler.
Okay, ma'am. Thank you.
Specific kind of markers are required for writing on cryo vials.
Where we can get that, ma'am?
A number of companies like High Media, it is available in their catalog. High Media, Thermo Fisher.
Okay, ma'am. Okay.
You
Okay,
can
ma'am. Thank
specifically
you.
ask them for cryo markers.
Cryo markers.
Yeah.
Okay.
Okay, ma'am. Our next question is from Dr. Reena Mehra in the chat box. Any specific crop is notified, suggested for Dr. Devraj, can you please mute yourself? Hello? Dr. Devraj, can you mute, please? Okay, ma'am, next question is, any specific crop is notified or suggested for pollen collection, or any crop pollen can be maintained?
Basically, at our place, we are maintaining the pollens of lychee, mango, oil palm, coconut, and basically these are being done as per the requirement of different institute in discussion with our experts, whether it is required or not. If someone wants to conserve the samples of their particular crop or species, then they can contact us. We'll see. If it is required, it is absolute necessary to pollen conservation is not our priority, but if there is requirement, and then we can go for their conservation. Presently, we are conserving mango, lychee, and oil palm, different plantation crops pollen.
Okay, thank you, ma'am. Okay, another question is from Dr. T.N. Saha from DFR. How long pollen can be stored?
Theoretically, we can say that once cryopreserved, the viability is maintained for infinite duration. And at our place, we have tested the viability of oil palm pollen up to, I think, 20 years. And they were found to be viable.
I hope Dr. Saha got the answer. Are you here? Are you listening? Hello.
Okay, madam. He has to know. Okay, madam.
Okay, okay. Any other questions? Okay, another question is from Dr. Hridesh. Ma'am, how to decide whether the pollen conservation is a particular tree species is required or not? For pollen
Basically, the decision has to be made by the specific crop breeder and specific crop group. But if there is asynchronous flaring of a certain short duration, we want to conserve the pollen, then we can go for pollen conservation. Like when male and female structures are not formed at the same time, there is difficulty in pollination, then we can go for short-term conservation of pollens for that purpose. And if in any crop, we have found difficult that we are not able to conserve the other explant, like in case of mango. We have found it difficult. If you want to maintain the true type explant, then we have conserved the pollen itself. The certain kind of genetic material is conserved for future. Basically, but it has to be decided by specific crop group and specific crop breeders if there is requirement or not.
I hope that is the answer he has got. Any other questions if anyone want to ask? No other questions in the chat box, ma'am.
Okay. Actually, if we could have demonstrated them these techniques physically, that would have been better. I am feeling that it may be quite confusing. All the techniques, when we discuss the techniques theoretically, they look similar in some type. They look interrelated and similar. When we do the procedures practically, then we understand the technique better.
Yes, ma'am. But still, those videos that you put was very nice, and I think for those who are totally unaware of the techniques, they might get benefited. At least the introduction was there for these techniques. And in future, if we will get to organize in physical mode, that will be much better for us also, for interaction point of view also, and for participants also. So this was a very nice practical session, ma'am. And really, we also wish that if it would have been in physical mode, it would have been more better. But still, we are very much thankful to you for providing such a nice and long lecture and covering all the techniques that are being followed in your division at our NBPGR in Gene Bank. So this is a great service that we are providing through you to conserve the rare germ plants. So thank you so much for being with us. And we hope that in future also we'll be able to organize such trainings and we'll get to know more about these techniques. Thank you so much, ma'am.
Thank you so much, Dr. Mamta, for providing us this platform to share the work which we are doing at our pace. Thank you. Thank you so much.
Thank you.
Shall I leave the meeting now?
Yes, ma'am. Sure. Yes.
Okay, thank you.
So still 15 minutes are there for the next session to start. So I request please be available. Our next session will start soon. Hello.
Again. And okay, thank you. Thank you very much. Thank you.
Good evening, sir. Very good evening, sir.
I'll just check. Hello. Good
Hello,
morning.
yes, sir.
Dr. Mamta, are you able to hear
Yes,
me?
yes, sir. Yes, sir. Yes, sir. You are audible, sir.
Hello.
Yes, sir. Yes,
Yes,
sir.
sir. Audible, sir. We can hear you, sir. Namaskar,
Hello.
sir. Hello.
Sir,
We-
we are able to hear you, sir.
Power on. Aux in mode.
Hello.
Hello. Yes, sir.
Madam, can you
Sir,
hear me?
sir, yes, sir. You are audible, sir. We can hear you,
Okay.
sir. Sir,
Okay.
good evening, sir.
Good evening, madam. Good evening, everyone.
Thank you so much, sir, for joining. And before I start, sir, I just want to give you a brief introduction of our participants before we start. So would that be okay, sir?
Okay, so should I introduce myself? Would it be okay?
I think before
I'll do it quickly.
No, no. Actually, what I'm saying is that before you introduce yourself, I would like to give a brief introduction of our participants.
Okay, please.
Okay, sir. Thank you. So, sir, in total we have 84 participants covering a total of around 19 states. Even we have participants from Leh Ladakh also, Jammu Kashmir also.
Great.
Then, if we talk about the institutions, so we have been able to cover around 39 institutions of different SAUs, then ICAR, PC units, KVKs, all representations are there of the participants. And designation wise, if I talk, so most of them are assistant professor and head, then plant breeders, research scientists, then technicals, so technical officers. So this is the audience for today's session, sir. Means for this 21 days training. So was the brief introduction, and now you can start, sir. I welcome you on behalf of all the organizing team, PGR organizing team, and NBPGR as a whole. And thank you so much for providing such valuable time to us from your busy schedule. Thank you so much, sir. Please introduce.
Thank you. Thank you very much, madam, and good evening. Good evening to all the participants. And it's really heartening to know that totally 84 participants are there. I don't know how many would be like, how many are there at the moment? Some 52 are with us.
Yeah.
So I'll give a brief introduction of mine. I am Dinesh Kumar Agarwal. I am working as a Registrar General with Protection of Plant Varieties and Farmers' Rights Authority. Basically, I'm an ICR scientist, and before principal, as a Registrar General, I was working as a principal scientist plant breeding with National Research Center for Banana. Basically, I'm a plant breeder, and I have worked in a number of crop species like fodder and forages, soybean, and cotton. And in between, I headed the institute that is meant for coordination of seed production and seed research. You might have heard of it. That is Indian Institute of Seed Science, MAU. So there I was officiating director for 4 years. So before I get on with my presentation, I would like to thank NBPGR for giving me an opportunity with so many young colleagues. And as you told that many of them, they are plant breeders, so probably they would appreciate and they would understand what I am going to talk for next 40-45 minutes. So I'll start sharing the presentation, and so that we can have it. So are you able to see the screen?
Yes, sir.
Okay. So you might have heard of, all of you might have heard of PPV FRA, and maybe some of you might be involved with dust testing. I don't know if any one of you is involved with the dust testing. So what we basically do today, I am here to talk to you about the protection of farmers and community rights on indigenous genetic resources. So how do we do it? And what this authority...
Excuse me, sir. Sorry
Yes?
to interrupt you. Sir, is it possible to put the slides in presentation mode?
I have put it in presentation mode only.
No, sir, we are not seeing it in presentation mode.
I have put it here at my end. I have put it on presentation mode only.
Okay.
Are you able to see? Like it is in full screen presentation mode only.
But we are not able to see, sir. It's still in slides. All the slides are visible actually.
Okay, let me do one thing. Okay, how come? I may stop sharing it, and then again I'll share. Is it okay this time?
Yes, sir. It's okay now. Thank you, sir.
Now it's in presentation mode. Okay, thank you. So now I'll begin my presentation. I'm going to talk to you about protection of farmers and community rights on indigenous genetic resources and how we safeguard. And before that, I would, in fact, like in my presentation, this aspect also would be covered. And I'm going to talk to you about what this authority does. Sorry. So you might be knowing what actually intellectual property rights are. Intellectual property rights are legal rights which are granted for a fixed period to exclude others from imitating, manufacturing, using, or selling a protected material or process. So you know that there are a number of intellectual property rights I have listed here in the table. Most of you must be aware of Patent Acts. Then there is the Indian Copyright Act, there is a Trademark Act, the Design Act, then Geographical Indication Goods Act, which you call GI, the Semiconductor Integrated Circuit Layout Design Act 2000, and lastly, the Protection of Plant Varieties and Farmers' Right Act 2001. This is the only IPR which is with the Ministry of Agriculture. All are dealt by Department for Promotion of Industry and Internal Trades. which is with the Ministry of Commerce and Industry. The last one, this authority, protection of plant varieties and farmers' rights, that is dealt by the Ministry of Agriculture. So why we had to introduce, you know, in a very idealistic world, in a utopian society, there cannot be a place for India. Somebody who invented the wheel, somebody who found out the way to conserve fire, they never got anything. So in an idealistic world, there is no place for it, but we do not inhabit such utopian world. So how it happened for last 150 years, continuously there has been a demand that even the plant breeders' right should be honored. So in 1991, you all must have heard about WTO. When we became a member of WTO, there was a requirement as per the TRIPS, that is trade-related aspects of intellectual property rights, under the Article 27.3, that we had to bring in either a patent system for the protection of plant varieties, or we can devise our own system, a sui generis system, or any combination of these two things. So we were given 10 years of time. And we had to develop a system of our own, and we preferred the sui generis system for plant variety protection in our country. PPAVFR Act, as I told you, as a developing country, we were given 10 years of time. So we enacted our PPAVFR Act in 2001, and it recognizes the contribution of farmers in agrobiodiversity conservation and includes farmers' rights. With regards to this last point, our act is very unique because nowhere else in the world the farmers' rights are protected in a way we protect, and in a way we recognize the contribution, the farmers, those who have been the conservers of traditional land races, those who have provided the genetic diversity to plant breeders to further work upon. So our act has the provision for these things. As I told you, we became a signatory in the year 2001. So we had to pass an act, we had to come out with our legislature within 10 years, of which we did in 2001. And after passing this legislation in Parliament in 2001, rules were framed in 2003. Then this authority, is Protection of Plant Varieties and Farmers' Right Authority, that was established in 2005. And first set of crops, because before we go ahead with the registration of crop varieties, we had to develop the dust pest guidelines. So the first set of crop notification, and for that, crop species are to be notified. Once we develop the dust pest guidelines, we would be notifying the crop species. Crop species, they become eligible for registration of new varieties. So first set of crops, which were 12 in number, they were notified in 2006, and registration of crop varieties. In actual sense, it started in the year 2007. So what are the objectives of this PPAVFR Act 2001, which was passed by our Parliament in the same year? It was to provide an effective system for protection of plant varieties, the rights of farmers and plant breeders, and to encourage the development of new varieties. If anybody who invests in plant breeding, if he is not able to recover his investment, then nobody would come forward. They would not put so much of investment in R&D. So we had to create an effective system which would protect the plant breeders' rights, which would protect the farmers' rights, and at the same time, which would protect the researchers' rights. And the second objective was to recognize the right of farmers. As I said, that our legislation is very unique in the way that we protect farmers' right, and we do it in respect of their contribution made at conserving, improving, and making available plant genetic resources, as I was talking to you. Because when we talk of innovation in the field of agriculture or in anywhere else also, always it happens to be incremental innovation. It doesn't have to be, it is not like a song, it is not like a novel, which is a creation of our mind. What we do is, when we breed a variety, always we require germplasm, always we require the basic building blocks. So the innovation always happens to be incremental in nature. So in that regard, with regards to that, we have to safeguard our farmers' rights for their contribution, which they have made in terms of conservation, improvement, and making available the plant genetic resources to the private or public plant breeders. The third objective was to protect plant breeders' rights, to stimulate investment for R&D, and if plant breeders' rights are not protected, nobody would come forward to put their money in R&D. So this was also an objective. To facilitate the growth of seed industry. If there is an investment in the R&D, then only the seed industry in the country would further grow, and it would provide farmers the good quality seed at a reasonable price, and at the same time, it would create so much of employment. So our PPVFR Act, it includes rights for researchers, it includes rights for breeders. Pardon? Hello. And then it's do you have any point, madam? Okay. If there is no point, so our act, I mean, it provides, there is a provision for researchers' rights, there is a provision for breeders' rights, there is a provision for farmers' rights. Now, this is in a nutshell, what and all are the component of our PPVFR Act 2001. As I said, there are researchers' rights, there are farmers' rights, there are breeders' rights, there are farming community, there is a provision for gene fund from which we give the rewards and recognition to the farmers I would be talking of. There is a provision for benefit sharing. If somebody has used the material that has been conserved by the farming community or farmer, they have to share the benefit which accrues out of commercial gain which they gain from that particular variety. Then there is a provision of compulsory licensing. By compulsory licensing, we mean like if any company or an institution has got his variety protected with PPVFR authority, and in future, if they fail to produce seed of that variety, PPVFR authority has the right to give the license to the third party, so that farmers would be getting the seed of that protected variety. So this is called compulsory licensing. There was a provision for appellate tribunal. Of course, it has been abolished now, so it is not there. Then there is a plant variety journal. Like if you have visited PPVFRA website, you would be knowing that every first we come out with a plant variety journal, which enjoys the status of gadget. And here, whatever variety we are going to protect, we publish the data in the plant variety journal. And the people, those who have the benefit sharing claim, they can always approach us. If there doesn't happen to be any opposition within 3 months, then only we give the protection to a variety. So this is the plant variety journal. Then this act provides, there is a provision for penalty for infringement for offenses. Then in the act, there is a provision, we have established dust centers where we conduct our tests for different crop species as per their guidelines. Then there is a provision for convention country. If we happen to be in a bilateral agreement with another company, with another country, then we can share their dust test data. Then even for the allotment of data priority, if they have applied in that country on an earlier date, in our country also, we would be taking it as a date of application. So all these provisions are there. Our act is very comprehensive. It covers all the aspects. Now I'll be talking to you one by one about all these. So there is a definition which is given for a plant breeder. So how would we define a plant breeder in legal terms? A breeder is who has bred, evolved, or developed any variety. While farmers are recognized as cultivator and as a conserver, and most importantly also. Farmer can register his own variety. So he is recognized as a breeder also in this act. There is a dual IPR protection in our act. PPVFR Act protects denomination of the variety as well as the variety and grants dual IPR protection. What does it mean is nobody, like suppose if a company has registered a variety with us with a particular name, with a particular denomination, nobody else can use that denomination for registration of their variety. And the second protection is no one can sell the variety which is protected with us with some other name. So protection happens to be dual in nature. Name is also protected and variety as such is also protected. So what are the plant breeders' rights? So plant breeders' rights with respect to protection of plant varieties are, they are the statutory rights created by and governed by the PPVFR Act 2001. And rights are right to produce, right to sell, right to market, right to distribute, right to import, and right to export. So very comprehensive set of rights are given to plant breeders. So breeders' rights, like if you had an opportunity to go through the act, it is defined in the section 28(1) of PPVFR Act 2001. It is an exclusive right to the breeder or his successor or his agent or licensee to produce, sell, market, distribute, import, or export the variety. It's very important. The word exclusive right. So nobody else can do it apart from breeder himself or his successor. his agent or licensee. Nobody else can produce, sell, market, distribute, import or export the variety. A breeder may authorize any person to deal with a variety registered under this act. They can sign the deed of assignment and they can authorize any other person to deal with it. And breeder shall enjoy the provisional protection. What happens is when someone submits the application with us, because we do not give the protection unless the variety has gone through the dust test, and it takes time. It takes time because for dust testing would be for 2 years. So most of the time it may take 3 to 4 years to grant him the final protection. But from the date of application till we grant him the final protection, breeder shall enjoy the provisional protection. That provision is also there in the Act. Then the second set of rights are researchers' rights. This is defined in section 30 of the Act, and use of any registered variety for conducting experiment or research. Suppose a particular breeder has registered his variety with us, he has got it protected, but still the other plant breeders, still the other researchers, they can use that variety for conducting experiment or research. There is as such no restriction on that. Then use of a variety. This again is a very important, the second point. Use of a variety as an initial source of variety for the purpose of creating the other. He can use the variety which has been protected with us as a parent in his breeding program without any restriction. The only restriction is he should not use it repeatedly. So this is the provisions. These are the rights which are available to researchers or plant breeders. Then farmers' rights. As I said, that our legislation is very unique in terms of number of rights it gives to farmers. So what are the farmers' rights? A farmer can save, use, sow, resow, exchange, share, or sell his farm produce, including seed of a protected variety. There is no restriction, because you know that in our country, like if I talk in terms of seed system, there are two kinds of system. One is formal system, one is informal system. Informal system is where farmers, they use their own seed for growing the next generation crop, or they would be exchanging seed among themselves. So majority of the seed which is sown, it to be the farm saved seed. So considering this very basic fact of our Indian agriculture, our farmers have been given these rights that they can save, use, sow, resow, exchange or share, or even sell their farm produce, including seed of a propagated variety. But there is a restriction, there is a condition. However, the farmers are not entitled to sell the seeds of registered variety in a branded manner. That is the only restriction. Otherwise, they are free to use it in whichever way they want to use. So there are a number of farmers' rights which are given in this act. As I was telling you, it is very comprehensive. Farmers enjoy so many rights in our act. First one is benefit sharing. I told you benefit sharing is if any material that has been conserved by farmers is used by a public or private sector institution in development of a variety, and if they earn money out of it, then they have to share the benefit with the original conserver of that particular material. Here in this case, farmers or farming community. So there is a major right. This is a major right which has been given to farmers. Then there is a provision of compensation. Compensation means if a variety that has been protected by us, if it is purchased by farmers, if it is grown by farmers, and it fails to give the expected performance, then that company or institution who has registered the variety with us is liable to pay the compensation to the farmers. That is the second right. The third right is reasonable seed price. It's not that any variety which is protected with us, any private company or public sector, they can charge an exorbitant price for that seed material. So authority has every right to put a reasonable price tag on the seeds of the variety which are protected with us. The next right which is available to farmers is farmers' recognition and reward for contributing to conservation. We know, as I was telling you, that plant breeding, basically it is the incremental innovation. What a plant breeder would do is he would be using the building blocks in terms of whatever germplasm is available with him. And as per the need, he would be making crosses, he would be creating variability, and then he would be selecting the material which is required. So every time for a plant breeding exercise, there is a building block, there are genetic resources which are required. And there is a provision in this act for farmers to recognize and reward them for their contribution in conservation of these valuable genetic resources. This is again a very unique provision which is there in our Act. I'll be talking about this, that in what way we recognize and rewards, how many rewards and recognitions are there, that I'll be showing to you. Then registration of farmers varieties. Here again, this class of varieties, farmers varieties, it doesn't exist in other legislation. We have given this particular to the farmers, that the material that they have been traditionally conserving or where they have made further improvement, they can register with us as a farmers varieties. Then in case of EDVs, development of essentially derived varieties, if any of the farmers material is used, there is a prior authorization which is required from the farming community or individual farmers who have been the conserver of that particular variety. Then exemption from registration fee of farmers. If you want to register a variety with us, it comes at a cost. One has to pay the fee, registration fee, one has to pay the dust test fee. But in case of farmers varieties, they are exempted in any fee. And then there is a very important protection which is given to farmers under this Act, that is innocent infringement. Even if a farmer has sold the seed, his farm produced as a branded seed, there is a provision if farmers is able to establish that whatever infringement happened, that happened out of ignorance, he was unaware of that, there is a provision to safeguard them under this particular provision, which is called innocent infringement, protection from innocent infringement. So basis of farmers' rights, as I'm telling you that there are a number of farmers' rights which are given in our Act. So why were we required to keep those provisions? Because you know that most of our landholding, they belong to marginal and small farmers. And those marginal and small farmers, I was talking to you in terms of the seed system, basically they raise the next crop by their own farm saved seed. So we have to keep this provision in our Act where so that farmers can use, reuse, sow, resow, they can exchange the seed. This is the very basis why we had to include the farmers' rights. Then again, a farmer in this Act, farmer is recognized as a breeder. As I told you, farmers, those who are conserving the traditional variety and where they have made the improvement also through selection, they can register that variety as a farmer's variety. That provision is there. You know, many of you would be knowing that last year, as I was talking to you about farmers' rights, so this is an important point I have to tell you. You all must have heard about the ITPGRFA, that's the International Treaty for Plant Genetic Resources for Food and Agriculture. This was signed on 3rd of November 2001. There are 147 countries. And in this treaty, in Article 9, there is a provision, like as you can read out from the screen, I put it, that the contracting parties recognize the enormous contribution that the local and indigenous communities and farmers of all regions of the world, particularly those in the centers of origin and crop diversity, have made and will continue to make for the Hello? Somebody is talking in between. So as I was telling, there is a provision in Article 9 of this particular treaty where all the contracting parties, they have to honor the contribution made by farmers, the indigenous communities in conservation and development of plant genetic resources. As I told you, there are 147 contracting parties, but hardly this provision has found a place in their national legislation. Our legislation is very unique in this sense that we have recognized the farmers' rights in multiple ways, and we give recognition to their efforts, particularly for the conservation and development of plant genetic resources. Last year, we had the ITPGRA governing bodies, the ninth version of that governing body meeting from 19 to 24. And there also, the basic theme was celebrating the guardians of crop diversity towards an inclusive post-2020 global biodiversity framework. And again, their farmers' rights were reiterated, and our efforts, the way we are recognizing farmers' rights, they were very well appreciated. And in this year, 2023, again, we are going to have one global conference on farmers' rights to educate the world how the farmers' rights are not in contravention with the plant breeders, right? How both the things can coexist. So this year, again, we are going to host a symposium to highlight the farmers' rights. As I was telling you, I was talking to you that there is a provision for honoring for farmers for their contribution for conservation of plant genetic resources. So there is a national gene fund. There is a provision in our act for creation of national gene fund. It is given in the section 45 of PPVFR Act 2001. And here we receive money from contribution from national international organization, annual fee paid by the way of royalty, then whatever benefit sharing is received, that also becomes part of gene fund, and compensation amount. As I was telling you that in Act, there is a provision for compensation. So whatever money we receive through these sources, they become part of National Gene Fund. And from this, we have instituted a number of awards like Plant Genome Savior Award. Then there is a reward. Then there is a recognition, which is defined in the section 39(1). And then this money can be used for strengthening panchayats in carrying out PGR conservation and sustainable land use activities. And this money is to be used in terms of benefit sharing. If there happens to be claim, if any farmer or farming community, they come forward and they are able to establish their claim that their material has been used in the development of a particular variety, which was protected with us, then the benefit which accrues out of that is shared with them. And then the money which is there in the gene fund, it would be used in paying the compensation to the farmers. If a particular company whose variety is protected with us and it fails to give the expected performance, then farmers can come to us to claim the compensation, and that money is paid out of this fund. So as I was telling you that there are basically three ways, three different awards which we give to the farming community. The first one is Plant Genome Savior Community Award. So it was stated from year 2009, and so far we have awarded 35 communities. Then there is a Plant Genome Savior Farmer Reward. So far we have given 51 rewards. Then there is a Plant Genome Savior Farmer Recognition. So far we have given 80 recognitions. And in case of Plant Genome Savior Community Award, it carries a citation, a memento, and a cash prize of ₹1 million. I tell you, this is the biggest prize money which is given by any government organization. Cash prize of 10 lakhs, and every year a maximum of 5 Plant Genome Savior would be given. In case of Plant Genome Savior Farmer Reward, it would be every year the maximum we can give 10, and it would carry a citation, a memento, and a cash prize of rupees 1.5 lakhs. And in case of recognition, it would be 20 in a number. The maximum number could be 20 in a year, and it carries a citation, memento, and a cash prize of rupees 1 lakh. So totally there is a provision to give the different award, rewards, and recognition to the tune of rupees 80 lakhs per year, which is a very huge amount. So far I was telling you about the farmers' varieties. Now, what and all different kinds of varieties are registrable under this act? There is a category which is called new variety. Then there is a category that is called extant variety. Extant means, dictionary meaning would be existing, the ones which are existing. Again, in extant variety, there are 3 subcategories: varieties which are notified under Seed Act 1966. Many of you, those who are working in active breeding programs, you would be knowing when you come out with a promising genotype which is established, which has been made homozygous, it is contributed for AICRP testing, and after multi-location, multi-year testing, there would be a varietal identification committee which would be identifying the varieties which have consistently outperformed the check, which are having all of the desirable features. And once the variety is identified, it would go to either to the Central Varietal Release Committee for formal release, or it would be released through the State Varietal Release Committee. So all these would be gazette notified by Government of India. So all those varieties which are gazette notified under the Seed Act 1966, they can be registered in an extant variety category. Of course, this provision doesn't happen to be available perpetually. Whenever we notify a crop species, we define a time window within which the extant variety can be registered. Because this particular category, extant variety, it is not there in any other legislation, particularly in UPO. It is all the time about the protection of new varieties of plant. As you would be knowing, the expansion of the term, that is the International Union for the Protection of New Varieties of Plant. So there is no provision for the registration of extant varieties, because Our country, we are the center of origin, we are the center of diversity for many crops. Many crops are our native crops, where farmers are conserving so many land races, so many traditional varieties. For that only, we have kept this provision for the registration of extant varieties, but it doesn't happen to be perpetual. Whenever we notify a crop species, there would be time window within which these extant varieties are to be registered. And within extant varieties, I said that there are farmers variety, I have already told. Then the varieties of common knowledge, which is known to the public, which is there in the common knowledge. These also can be registered as extant notified varieties. Then essentially derived varieties. Essentially derived variety also, which would be derived from an initial variety while retaining all the essential characteristics except that they were developed. So farmers variety, again, like it has been defined in our act, the variety which have been traditionally cultivated and evolved by farmers in their field. It could be a wild relative or land variety about which the farmers possess the common knowledge. And as per the provision under section 2J, 15.2, farmers varieties are protected as extant varieties, which I showed you in the previous slide. So what are the eligibility criteria for the registration of a new variety? A new variety has to fulfill 4 conditions. The very first one is of novelty. The second one is of distinctiveness, uniformity, and stability. And I'll deal with each one of these 4 criteria one by one. A variety would be called to be novel if the propagating or harvested material of the variety is not sold or otherwise exploited for. The condition for 2 different group of crops is given. For trees and wine, it should be less than 1 year in India. And outside, if it is known for less than 6 years, it would be considered novel. Apart from other than tree and wine, in India, if they are not been in commercial use, if they are not known for less than a year, if they are known for less than a year, they would be considered, they would fulfill the condition of novelty. And abroad, it should not be older than 4 years. It should not be beyond 4 years that it is in commercial use. It is in trade. So if it is less than 4 years, it would be considered novel. What is distinctness? The slide itself says you can very easily discern like how every single fruit is different from every other fruit. So, like as I told you, when we notify a crop species, there will be that list of dust characters. So if any variety which is distinct from any other variety which is there in the common knowledge, which is there in the database, public database, then it would be called distinct. What would be a uniform variety? Again, you can see, I don't have to explain. I think it is very clear from the slide itself. What is a variety which is having uniformity? What is a variety which is not having uniformity? What is a stable variety? Again, the slide says very clearly, the one which is able to maintain its performance over the repeated propagation, that would be called stable. If there happens to be variation over the repeated propagation, it would be considered unstable and it would not qualify for registration as a variety with the PPBFR. So this is the basic process how we go about when an application comes under what and all different stages it has to go through before it qualifies for the registration. And there are different provisions. So I have mentioned it here, and maybe I'll make available this particular slide so that you can go through. And a denomination. What are the restrictions? Denomination. Like while naming a variety, when an application comes, the first thing before allotting the registration number, we check whether the denomination is right, it is acceptable or not. And there are a number of conditions for that. It should not consist solely of figures, maybe letters or combination of letters and figures. It should not just be the numbers alone. It should not hurt the religious sentiment. We cannot keep a name of a variety which would hurt the religious sentiment of any community. We have to avoid it. Should not be a name or emblem. Should not be comprised solely or partly of geographical name. That again is not acceptable. Registrar may allow it if it is based on honest use of such denomination. That liberty, that discretion is available with the registrars. Then it should be single and distinct, and the denomination cannot be a trademark. So these are the restrictions while deciding whether the denomination or the name of the variety is acceptable or not. Then how do we do the dust testing, which is defined in the section 19, and it is for your knowledge. The dust test shall be field and multi-location based for at least 2 crop seasons, and there would be a special test if the dust establish a distinctness. The dust test for varieties about which there is a common knowledge will be for 1 crop season at 2 locations. For the new varieties, it would be for 2 seasons at 2 locations. But for the varieties which are of common knowledge, it would be for 1 crop season at 2 locations. For farmers variety, a grow out test will be conducted for 1 crop season at 2 locations, and the uniformity level of farmers variety for the respective species shall not exceed double the number of off types such as may be specified in the journal. Like for seed production, specified the number of off types which are permissible. So in case of farmers variety, there is a relaxation, but still it should not be more than double than what is there for the seed production. Then manner of testing essentially derived varieties, it is decided case by case by the authority on case-to-case basis, and fee for dust test to an extent of maximum of rupees 50,000. Whenever we develop a dust test guideline to the centers to which we allocate the project, we would be asking them what would be the expenses in raising the trial for 2 years. Based on that, we decide the dust test fee, but there is a cap on that it can be maximum rupees 50,000. This is for the off-site testing. For on-site testing, it can be more than 50,000. So, where do we carry out our dust testing? For that, we have the designated test centers across all crop species which have been notified. And as you can see, so far we have notified 180 crop species. There are about 153 dust test centers. There are dust test centers which would deal with the multiple crop species. Otherwise, you would be wondering why the crops are 180 and dust test centers are lesser than that. That is the reason. So totally locations are 153. And there are a number of activities which are carried out at dust test centers. They carry out dust tests, and then they have this responsibility for maintenance of example and reference varieties. They would be doing the characterization of varieties in reference collection or public domain for updating the database. And they would conduct the on-site testing by recording data of candidate varieties. Suppose in case of horticultural crop in a tree species, in an ornament, in an orchard like mango. So it cannot be brought to the dust test center. So what we do, the applicant would be having the site, and the dust test center would facilitate the recording of observation at applicant field. So as I was telling, in case of vegetatively propagated crops, planting material has to be submitted to respective dust test centers as per the details available in dust test guidelines. And there is a provision for the on-site testing also. And applications for farmers varieties, they shall be forwarded. Like there is a provision. There is a provision when a farmer he wants to register the farmers varieties, he has to get his application forwarded by the concerned panchayat officer to SAU or ICAR institute before the submitting the application for registration. Then crop specific breeder of that SAU in whose jurisdiction that farmer's area is, or ICAR institution, they would certify the seed material for its authenticity, purity, and uniformity by conducting 1-year testing that they would be doing. And there are standing instructions from authority to SAUs and ICAR institutions to carry out this exercise. And further, the application shall be countersigned and forwarded to the authority by Director of Research of SAU or Director or Principal Coordinator of ICAR institutions before the onset of next season as described in protocol for receiving the application. As I told you, the very first step when we start registering the plant varieties are development of dust test guidelines. So we would be inviting project proposals from different institutions, and we would be giving them money. We would be providing them facility in terms of manpower and other infrastructure facilities. And then they would propose the list of characteristics which can be used as a dust for the dust guidelines for the dust testing. And then we would constitute a task force committee. There would be a number of meetings. We would finalize the dust test guidelines. It would be approved by the authority. Then it would be further forwarded to the ministry for the gazette notification. And once these dust test guidelines, once the species is notified by the ministry, then only the new varieties of that species can be registered. Of late, we have come out with a public notice, particularly for the farmers' varieties and varieties of common knowledge, that even if a crop species has not been notified so far, but if a farmers want to register his variety in that species, or if somebody wants to register the variety of common knowledge in that species, they can still come forward, they can make an application, they can provide the minimum data that we have given as per, so that we would be having the varietal description. And those varieties, farmers' varieties and varieties of common knowledge, can still be registered with us, even if we have not notified the crop species so far. This is the very recent change, and we came out with a public notice in 2022 only. So tests to be conducted, we conduct the dust test, that which happens to be the field test, multi-location for at least 2 crop season. Dust test, it would generate an official description of the variety, which is very essential. The special test would be conducted if dust test fails to establish the distinctiveness. And for trees and vines, as I told you earlier also, there is a provision for the on-site test. So this is the list of 180 crop species which are notified where the new varieties can be registered. And as I told you, again, I would repeat, even if a crop species has not been notified so far, and if a farmers want to register his variety, or somebody wants to register the variety of common knowledge, they can still come forward and make application, even for those species which do not find a place in this list. So criteria for registration already I have defined, I have told you. And now what would be the period of protection? So for trees and vine, it happens to be for 18 years. Initially, it would be for 9 years, and then one has to get the renewal, which can be further for 9 years. So total duration would be 18. In case of other crops other than trees and vines, it would be totally for 15 years. Initially, we give the protection for 6 years, which can be renewed for another 9 years. In unnotified varieties, it would be 15 years from the date of notification of variety under the Seed Act 1966. So suppose a variety is notified in the year 2019, it would be 15 years from 2019, because that would be the total duration. So this again is the, I mean, for how many years, for at how many locations we would be doing for the different category of varieties. Already I have told you, so I can skip this slide. So this dust test also we have talked about. Now in terms of, this is all about Act, how we do the registration. So how we have progressed over the year. As I said that we started in real sense the registration of crop varieties in the year 2007. So from 2007 till 2nd of February 2023, we have received totally 17,971 applications. I have given the year-wise breakup. You can see a few years preceding to this year, there has been a reduction because of COVID, and there were other restrictions. For that, there has been number has dropped down. Now the 17,971, if you to consider in terms of application wise, so you can see 60% of the applications which we have received so far, they have been from farmers. So how effective it has been for safeguarding the farmers' interest. You can see it from this slide. So out of 17,971, 60% of total applications which we have received so far, they are from farmers. So how effective our system has been for safeguarding their interest. Now again, if I have to put it in terms of applicant wise, so mostly 60% are from farmers, and nearly 4,462 applications have been from private companies, and half of that, nearly 2,327, they are from the public sector institutions. So this is the total number of certificates which we have received after the dust examination and after they have completed all the requisite requirements. Totally, we have issued 5,011 certificates. This is the category wise. You can see out of 5,011 certificates which have been issued, the highest is for the farmers varieties, followed by the extant notified varieties, which is the Seed Act, followed by the varieties of common knowledge. I mean, followed by the new varieties, and then the varieties of common knowledge. So this is the number of certificates which we have issued for different categories of varieties. You can see Mostly it is for the farmers varieties, and essentially derived varieties we started only from the year 2012. So far only 14 certificates have been given, but in case of extant notified varieties, again a number of certificates have been issued, totally 1,437. And similarly, extant VCK, there have been 688 certificates which have been issued. This again is applicant wise, like how many are from public, from private, and farmers. So in summary, protection of intellectual property is extremely important. As I was talking to you, you yourself must have realized, for supporting and driving innovations, as I said, that nobody will be putting money if plant breeders' rights are not recognized in any way. While safeguarding the farmers' rights, we have to recognize the plant breeders' right. Then only there would be an investment, there would be further R&D for the varietal development. The plant protection, it encourages and promotes plant breeding activities and export, which is understood. Indian plant variety protection law is unique as it simultaneously aims to protect the plant breeders as well as farmers' rights. I've already made a case, I already made a presentation for that, how we safeguard the farmers' rights. The analytical review and registration of horticulture crop varieties indicate that the private seed sector has taken keen interest in protecting their proprietary varieties in comparison to the public institution. This is true, particularly for the horticultural crops. India is one of the center of origin for many horticultural crop species of economic importance, and it is essential to protect the plant genetic resources in the farmers of farmers varieties through IP right, not just for the horticultural crops, even for our agricultural crops, which constitute the backbone of food security. And so we have to promote. I would appeal to all of you, since you are there from the different institutions, you have to map the farmers varieties, which and all are the farmers varieties available in that your area. You have to encourage them to come forward for registration with PPVFRA, and at the same time you have a role to play in making the material uniform, in developing the varietal description for those varieties for which we all the time we provide financial assistance. So I would expect and I would request to all of you to kindly take a keen interest in it and facilitate the farming community, all the members of our farming community, to protect their IPR, particularly for the farmers varieties. So this was in nutshell about PPVFRA. It's over from my side. Dr. Mamta Singh.
Yes, sir. Yes, thank you so much, sir. And this is really one of the burning topic as well as evergreen topic in for PGR management.
And just now, madam, I'm seeing Dr. R.K. Gautamji here.
Yes, sir. Yes, sir. He is there.
Good evening, Dr. And I was telling that, like for registration of new varieties, there is a restriction that crop species is to be notified by the ministry. It has to be gazette notified. Then only we can accept the application for the registration of new varieties. But no such restriction exists for farmers varieties or varieties of common knowledge. So recently, as you sent me a mail for Gadmal, which is like urad, but may not exactly be urad. So even if it happens to be the species which is not yet notified by ministry, we can still register the farmers varieties and varieties of common knowledge. So because today's topic, it was about how to safeguard our indigenous plant genetic resources. So this I wanted to tell you. So once you send us the basic description of that Gadmal, whether it is urad or not, even if it is not yet notified, we can go ahead with the registration. This I wanted to tell you, and to all the participants, I wanted to drive this point.
Sir, there are few questions in the chat box.
Okay, madam.
So,
let me see. Okay, let me see. What is the deed of assignment while submitting the proposal of registration? Deed of assignment is when you are transferring the right, when you are a breeder. Like if you are from ICR institution, basically you would be signing the deed of assignment in favor of director. So he would be making further correspondence. The institution would be having the right over that variety. That is the deed of assignment. And what is the fee for the registration? The registration fee would vary for the different category of varieties, like whether you are a public institution, whether you are a private company, so registration fee would vary as per the category to which you belong to. As if you visit our website, you can find the detailed information on that what fee is available in your case. That is one thing. And the dust test fee it would vary from crop to crop. So there are number of components of the money you have to spend. The very first thing is registration fee, which I told you that it would vary depending on which category of applicant you fall into. That would be there. And the dust test fee, it would vary which particular crop species you want to register for. It would vary from crop to crop. And in case of, as I told you, that in case of agricultural crop, there is a cap of maximum rupees 50,000 which we can charge for the dust test fee, but it may cross that limit in case of on-site testing. That is one thing. And if a variety releasing center is also a nodal center of dust testing, in case dust testing for 2 seasons from same center is acceptable. Yeah, there is no restriction, but it would not be the concerned breeder who would be doing the dust testing. Basically, we have given it to the seed technologists, those who are there in the ICER institutions who are developing the seed variety, but it's perfectly all right. And in many crops, we have multiple dust test centers. So we have this liberty also. So we may not assign the job to the concerned dust test centers. We can give it to other centers also. Then there is a question, what is the criteria of dust testing of species like Deodar? That is what, for a number of crop species, as I was telling you, there are in total 180 crop species which we have, which ministry has notified so far. We have developed the dust test guidelines so far for 189, but another 9 which have not yet been notified by the ministry. It has not been gazette notified. So once the species is notified, the dust test guidelines are available. So if for Deodar, I'm not very sure whether the dust test guidelines are there. You can always find it out, and I'll also go through it, whether it figures in the list of 189. If it is there, the dust test guidelines are there. If they are not there, you are interested in developing, you may submit a project with us. Then there is one question by AG. It was noted that the guidelines of characterization were made long ago, but with new varieties, many new characteristics have been introduced in crops. For example, in wheat, grain, it is categorized as wet. Okay, I got your point. And what I mean to say that dust test guidelines are not something which are sacrosanct. They can be revised number of times. In fact, recently only we constituted a committee for revising the dust test guidelines for wheat. Already 2 meetings have happened under the chairmanship of Dr. GP Singh, Director NBPGR, and very soon we would be putting it in our plant variety journal, the revised test guidelines. We are doing it for number of crop species, for maize, for pigeon pea and all. So as and when there happens to be a requirement, there is a requirement for revising it, we do it. So it's not that those are sacrosanct and once we are published, they cannot be revised. All the time that can be done. Sir, what will be the procedure of dust testing for forestry, agroforestry trees? As I told you, for number of forestry species, we have already published, we have already notified the crop species. So as per guidelines which have been notified, we are doing the dust testing. And we have a number of, for dust test centers there in IFGTB in Coimbatore, Indian Forestry Genetics and Tree Breeding Institute. They are the dust test centers. So if you send an application, we may provide the material to them. If you are interested in on-site testing, that also you have to ask for, and the dust test centers would facilitate that exercise. So I think these were the questions so far. Anything else is there?
Hello, sir.
Dr. Vikas Pawar is there. He has raised his hand.
Okay,
Sir,
you
can ask.
Okay, Pawar sir, please go ahead.
Sir, thank you, sir. Wonderful lecture. I have one query that as we have adopted, as per the TRIPS, we have adopted the sui generis system.
Yes.
Then how the BT sold by that Monsanto Meiko, I think it is a patented technology, BT technology. How it is, under
like
which rule it is sold in India?
So that is what we are not talking about. We are not into the regulation of seed business and all, isn't it? We are not in the regulation of seed business and all. That is with Ministry of Agriculture and Farmers Welfare, we are about the protecting the, if a person comes forward and he gets his variety registered with us, then nobody else can produce the seed of that particular variety until unless the original breeder assigns him the right. So what you are asking is that it is a patented this thing, how they can sell it. That patent is for trait, not for the variety. You know, as per the as such in our patent system, crop plants or any living plant or animal cannot be patented. It is the trait that was patented, and once they have entered into the agreement with any licensee, it can be an Indian company. They can sell it. They can sell it. There is no restriction on that. But suppose a variety which carries a particular gene or particular event gets registered with us. It gets registered with us, and if anybody else wants to sell the seed of that same variety, they have to take the permission. They have to get the license from the company which has registered that material with us. So our business is with the protection of it.
Hello,
Okay, sir. Thank you.
Thank you.
Hello, sir. Good evening, sir. Thank you for wonderful presentation, sir. I have one question.
Who is it? Okay,
Any
Dr. Ken Gupta. Yeah.
Sir, any criteria or limitation or restriction for registration of farmer variety for pest and disease data? Is it necessary?
What pest and disease data? Except like I am not able to fathom your question. What do you mean to say? Gupta sir.
Sir, any restriction or limitation for registration of farmer registration or our germplasm under pest and disease data, is it necessary? For disease and pest data?
I think I can supplement, sir, on this issue if you allow.
Yeah,
Yes,
basically like how you can register the farmers varieties. I told you, in case of farmers varieties, that has to come with a forwardal from the concerned director of ICR Institute or director of research of state agriculture universities. And then in what way university has to facilitate him is in purifying the material, because you know that the farmers varieties would be mixture of number of it could be pure lines. So you have to help the farmers in terms of purification. Then you have to develop the description of a variety. Once you do these two things for the farmers variety, you would be sending to us and we would find it out and we would give the registration. That is how it is. And in case of dust characteristics, there are no traits which are involved, which are associated with a disease or insect resistance, to make things clear to you. Hello. Dr. Gautam, you wanted to say something?
Yes, yes.
And it can be done if it is having the resistance, it can be done. There is no issue about it, but you have to develop the description for the dust characteristics.
Yes, sir, you are very right, sir. And my response to his question would be that if he's talking about disease or pest resistance, it has to be scientifically validated. Exactly. And as you mentioned that the researchers should help the farmers in purifying their varieties and then test at their regional, their research stations, this material for disease resistance, adopting the proper protocol for it. And they can also submit such germplasm to NVPGR also, because we register the traits specific germplasm. So that way it can be done. And if the variety, farmer variety is having overall merit, then it can be submitted to PPFRA for the protection. This is what I would like to.
And Gupta sir, I have a request.
Thank you very much, sir.
Dr. Gupta ji, I have a request to you. There has been number of farmers varieties application which we have received through JNKV Jabalpur. But what happened is still, like many times we have written letter, we did not receive any seed material. So you facilitated the submission of application. Of course, it may not be you. I am talking for the university only. So please, like in between, we have sent number of requests. So please go through that, and whatever information is lacking, you please provide that information to us so that we can go ahead with the registration of farmers variety. You please see to it, like I have written number of letters to Director Research. Okay.
Okay, thank you very much, sir.
If
yes, sir.
One more question is there in the chat box.
Okay, they are still coming. Devdar.
One question has come.
It was noted that guidelines of characterization were made long ago, but this already I have replied. Sir, what will be the procedure of dust testing? This also I have replied. Why the applications for registration of varieties from private and farmer sectors more, but public sector almost, what is it? Public sector almost half the number.
Half the number.
See, there are two things. For public sector, they would be coming out varieties which are notified under Seed Act 1966. And as I told you, in case of extant varieties, it is not perpetual in nature, because nowhere else in the world they recognize the varieties which are in existence. It is about the new varieties of plant. So when we kept this provision, we could not keep it that anytime you come and register the variety. There happens to be a time window. And as you would be knowing, if you have seen our public notices for 102 crop species, they are no longer eligible for the registration of notified crop varieties, which include many of important cereals, pulses, oilseeds and all. So no longer we are accepting applications for the varieties which have been notified under Seed Act 1966. That is one part. And second thing is, registration always involves some cost. So it is up to the applicant only. It is up to the applicant only. In case of public sector institutions, there are only few institutions like ICR which are licensing their varieties. So if they are not going to earn anything, if they are not going to accrue any commercial benefit of this, probably the maintenance cost, the annual fee, renewal fee may not be a very, very attractive proposition for that. That could also be a reason. And of late, we have, as I told you, the first thing, of late, we have stopped taking the application for number of crop species under the extant notified category. What is this seed quality test to be submitted is acceptable from any private company? While submitting the seed, they have to give the germination percentage, the moisture percentage that is there invariably for all. Whosoever applies for the registration along with the seed material, they have to provide this information. So it is there for all. Any other thing? I think there is nothing more in the chat box.
Yes, sir, there are no more questions. So since our head sir is here, Dr. R.K. Gautam sir. So sir, do you want to say some final words?
Okay. Thank you, Dr. Mamta. Sir, namaskar and good afternoon. And once again, our hearty welcome to our today's guest speaker, Dr. D.K. Agrawalji, who is the Registrar General PPFRA. As we all know, agrobiodiversity is very important for the sustainability of agriculture and humankind on planet Earth. But recognition, benefit sharing, and incentivization for promotion of agrobiodiversity conservation are also far more important. In this direction, we are very lucky to have PPFRA, which is a Protection of Plant Varieties and Farmers' Rights Authority in our country, to appreciate and incentivize the farmers and custodians of agrobiodiversity conservation. Sir, I, on behalf of entire organizing team and ICR NVPGR, and on my personal behalf, extend a warm vote of thanks and gratitude to you. You have befittingly and beautifully shared the latest updates on IPR issues, breeders, farmers' rights, and how PPFRA can facilitate our farmers and NAIRS system as a whole for agrobiodiversity conservation and IPR protection for breeders, farmers, varieties. I am sure all trainees have been richly enlightened on this topic, and there were several questions which Dr. Agrawalji has very nicely covered, so that our all trainees can also initiate efforts for enabling farmers in their respective places for variety recognition and PPFRA awards. I would like to mention that earlier I was working in Andaman and Nicobar Islands, which is one of the agrobiodiversity hotspots of our country. And our team could facilitate the one Crane rice community whose forefathers had migrated from Myanmar to India somewhere in 1925. And we could help them in not only in the registration of their rice varieties, but we could also help them in getting the Plant Genome Severe Community Award of rupees 10 lakh in 2016. So we are grateful to PPFRA sir for this generous support. Since NBPGR is also working on plant genetic resources, and we need your further guidance and support in our all the efforts in this direction. At ICR NVPGR, as you have kindly mentioned, we have initiated some work on Gadmal, a new type of pulses for its on-farm conservation, documentation, taxonomic and molecular profiling for facilitating the Gadmal growing farmers for these incentives and awards. We request your kind guidance and support for this initiative and many more from NBPGR. At last, once again, I, on behalf of organizing team and NBPGR, I heartily thank you for sparing your valuable time and sharing your knowledge and expertise on this topic. Thank you very much, sir. Thank you.
Thank you. Thank you very much, Daksa, for providing me an opportunity. Thank you very much.
Thank you.
And thank you to all the participants also for the patient listening. Thank you. Thank you.
Thank you, sir.
Thank you very much, sir. Yes, Dr. Mamta, over to you.
Yes, and now this was the last session, so I think we will wrap up for the day. Thank you, sir. Thank you, all of the participants and all the members. So we will start with tomorrow's session that will be starting from 9:30. Any updates are there, they will be updated in the group. So we'll be back tomorrow at 9:30.
Okay, thank you. Thank you very much.
Okay, sir.